Welcome, PA Class of 21. My name is Michelle Murphy, and I'll be your instructor as we go through human physiology and pathophysiology in these first two semesters. I'm excited to see you again next week. We've all met at interviews, but I'm excited to have you back on campus. Normally, this lecture is released when you get to campus, but it's a long one, so the class of 20 asked me to release it to you early. I was hoping to get it to you last week, but we'll get it out a week early, and hopefully that'll help a little bit. Mostly it's a review of undergraduate physiology, so it's longer. It's the longest of the semester, but hopefully a lot of it will be review. But then some, it may have been a while, or some components may not have been hit very hard in your undergraduate physiology. So I want to give you some time to go through it and maybe to brush up on some things that you may not remember quite as clearly as you'd like. So we'll go ahead and we'll jump right in. In this first unit, we're going through chapters 1 and 3. So it's mostly a review of cellular biology. Our objectives for chapter 1 will review the structure and function of cell organelles. We'll review the structure and function of the cell membranes. We'll describe the modes of cellular communication. We'll describe the major steps of cellular respiration to produce ATP. We'll compare and contrast the types of membrane transport. We'll describe the mechanisms involved in the generation and transmission of action potentials, and we'll review the cell cycle. So we'll jump right in looking at cell. Objective 1 will review the structure and function of the cell organelles. When I do these, a couple things about these lectures, I do them all as one go, and so they get to be quite long. However, they're broken down into objectives. I highly suggest that you just do one objective at a time and then take a break. When you can get into the Blackboard course, and I can't mail it out because it's too big, but when you get into the Blackboard course, the narrated PowerPoint will actually be in there, and you can use that to make an audio file, to break it apart into pieces, however you want to study it. By taking the raw narrated PowerPoint, you can organize it however you want. I will always render it as a one long video, but that does not mean my intent is for you to sit there and watch one long video. That's not a good learning environment. I would suggest doing one objective at a time and then taking a break, thinking about, reflecting on what you've learned, what's new, what did you already know, and then coming back and looking at another one. You're not going to be able to focus for however long these videos are, and I never know from the outset how long they're going to end up being. But it's best to break it apart into pieces, but you will have that raw PowerPoint, narrated PowerPoint, if you want to structure it a different way. That's up to you. When we look at cells, there are two main kind of big divisions of cells, prokaryotic cells and eukaryotic cells. What we're going to be looking at mostly are eukaryotic cells, but by contrast, the prokaryotic cells are the smallest, simplest cells. They have nucleic acids, DNA and RNA, but they don't have a nucleus. They do have ribosomes for protein synthesis. An example of prokaryotic cells would be the bacteria. The picture on here and what we mostly focus on are eukaryotic cells. Eukaryotic cells are larger, they're more complex, the DNA is arranged in linear chromosomes, and it's enclosed in a nucleus. They have not just ribosomes, but also mitochondria, an endomembrane system, and extensive vesicles, and we'll talk about all of those things. So eukaryotic cells are what we focus on in this class. The first organelle we'll look at is unique to the eukaryotic cells, and that's the nucleus. The nucleus has a membrane. It's a nuclear envelope here, and it has these pores, and these pores allow things like messenger RNA to leave the nucleus. But messenger RNA is too big to diffuse through a membrane, so by having these pores, substances can communicate between the nucleus and the cytoplasm. The outer membrane is continuous with the endoplasmic reticulum. We'll see that a little bit better on the next slide. Linear DNA, here we have linear DNA, and it's wrapped around histone proteins. Here's the histone proteins in order to form these organized chromosomes. That organization is necessary in order to go through the process of mitosis and to stay organized. So we'll look at that more in chapter 4. We'll talk a little bit about cell division in this chapter, but we'll come back to that when we talk about genetics. In the nucleus is where that DNA is transcribed to messenger RNA. So the DNA always stays in the nucleus, but we have to get the code from the DNA out to the cytoplasm in order for the ribosomes to use it. And so the way we do that is we make a copy of the DNA, the particular gene we're interested in, and that's called messenger RNA. If any of those genes are missing, then that messenger RNA code is going to be inaccurate. So if they're malformed, if they're missing, if they're mutated, then that code is going to be not valid. And that can have a pathophysiological outcome. For example, if there's a mutation in the genes that code for the clotting factors, that can lead to inherited hemophilia. And we'll see that there are a lot of those conditions where they're looking for those genes and have found those genes, and we know what's causing those pathophysiological conditions now because the genes have been identified. And that is an ongoing process where more and more genes are becoming known as far as what kind of pathophysiology they cause. The nucleolus here, they've got it kind of popped out. So here it would fit right in there in the center of the nucleus. The nucleolus is a small dense RNA structure used for ribosome assembly. As you go through these slides, if there are any terms such as maybe nucleolus that may be a while since you've seen it, you're not really familiar with what it is, or you can't just define it off the tip of your tongue, I would highly suggest making a flashcard. And this is because students have told me that it's helpful to make a flashcard on things you don't know, because your exams in this class are timed, they're multiple choice, and so if you have to sit and think about what a word means, you're using up some of your time. And so it's much more efficient if when you go through these and you're like, I'm not sure I could define that off the tip of my tongue, to make a flashcard and then go through those so that when it comes to the test, you can just go right through those words and you don't have to stop and think what they mean. Ribosomes are RNA protein complexes. They may be free or attached to the endoplasmic reticulum. If they're attached to the endoplasmic reticulum, that's called rough ER. Now here's the nucleus, and we saw the nucleus getting tongue-tied already in the previous slide. And here's the endoplasmic reticulum, kind of continuous with that nuclear envelope. And that goes all the way around three-dimensionally. And if here we have a cross-section through it, and you can see that on this part of the endoplasmic reticulum, we have these little ribosomes. And so that's called rough endoplasmic reticulum because it looked bumpy under a microscope, so they called it rough, and it has those ribosomes. There's also smooth endoplasmic reticulum that does not have the ribosomes, but that's still continuous, a part of the system there. The ribosomes are responsible for protein production. They are where that translation of the messenger RNA code into the amino acid code occurs. That occurs at the ribosomes. In the central dogma of biology, we know that the DNA is in the nucleus. So that's in the nucleus. But that code has to be converted into a protein in the cytoplasm. So we have to get it to the cytoplasm. And we do that by, in the nucleus, going through the process of transcription. Getting used to using I haven't done one of these lectures since November, so I'm getting used to it'll take me a little while to talk and write, get used back into the mode of that again. So through transcription, we generate messenger RNA. Messenger RNA then can go out through the cytoplasm to the ribosomes. It doesn't become ribosomes, it's going to the ribosomes. At the ribosomes, the amino acids, which are in the cytoplasm, will join to form proteins by translation of this messenger RNA code. So translation of this code at the ribosomes will take those amino acids in the right sequence, whatever the code says they should be in, and at the ribosomes bind them to form proteins using that sequence that was on the messenger RNA. Once those proteins are formed, they have to fold into their correct orientation, and any kind of cellular stress might disrupt that. That also, not just a mutation of the DNA, but also anything along this process, like any problem with folding those proteins, can also cause the cells to malform. Smooth endoplasmic reticulum is responsible for lipid and steroid production. So it doesn't have any ribosomes, but it is still producing things, and it's producing those lipids and steroids. Coming out from the endoplasmic reticulum, we have the Golgi apparatus, sometimes also called the Golgi complex or the Golgi bodies. It means all the same thing. Different books will use some of the different terminology, and so depending on what you learned in your undergrad, please realize that a lot of this terminology we may use an alternate term, but that's just because there's not necessarily agreement among all the undergraduate texts. So sometimes I'll even go back and forth between them, just depending on how I have it written, but all those mean the same thing. Golgi complex, Golgi apparatus, Golgi bodies, all the same for our purposes. The role of the Golgi complex is to finish the protein. So they were produced on the ribosomes, but they still have to be activated. So they have to be folded correctly. They have to be sometimes cleaved into their pre, pro, or active form, depending on what this cell releases. Does it release a preform like angiotensinogen, which is going to have to be activated in the cells, or does it release an active form of a protein? It just depends on what that gene was coding for. The Golgi complex will finish that process. Proteins are often secreted in a pre-pro form and activated by tissue or blood enzymes as they're needed. They're released then by the secretory vesicles. Here you can see the secretory vesicle. That's going to fuse with the plasma membrane and release its contents out into the interstitial fluid or into the blood. Lysosomes are vesicles filled with digestive enzymes. They digest unused metabolic products and worn-out cell components into simple carbohydrates, amino acids, and fatty acids for recycling. That process is called autophagy or self-eating, and it helps to maintain homeostasis so these worn-out cell components and these excess metabolic products aren't building up inside the cell and causing toxicity. They sense nutrients for metabolic adaptation. So sensing those nutrients allows the cell to coordinate the cellular output to match what the body needs homeostatically. So it's a very dynamic system where what is needed depends on, or what is provided depends on what is needed. And so it requires a lot of sensing and interacting. Any malfunction in that can lead to an accumulation of cellular debris to toxic levels. And there are known conditions, and I'll just give you a couple, that are related to these lysosomal disorders. One is Tay-Sachs disease. In this condition, a lipid, a GM2 ganglioside, accumulates in the neurons because the lysosomal enzyme that would normally break it down is deficient. That enzyme is produced by a gene mutation, the hexa gene mutation, and with that accumulation of those lipids, that disrupts the neuron function because the neurons can't clear them out. And so they clog up the neuron, and with those lipids, it can't carry out its normal ion channel action potentials, and it loses its function because the lysosomes don't produce the enzyme needed to break down those lipids. Another is gout. With gout, there's an accumulation of uric acid, and that uric acid accumulation in the lysosomes destroys the integrity of the membranes, and the enzymes leak out. And when those enzymes leak out, here's our lysosome, we should keep our digestive enzymes inside there to break things down. But if the enzymes leak out, that's going to cause inflammation in the rest of the cell. So it's going to get inflamed and it's going to be painful because those enzymes should stay in the lysosome and take care of things, but the uric acid is destroying it. Another note about these, whenever I have something in parentheses, it's usually to provide further information. When you're looking at the notes, don't memorize these. If I've got a gene or something in a parentheses. I don't want you to memorize anything in the notes. I want you to understand it, but don't focus on, if you're making flashcards, don't focus on the things in the parentheses. But definitely, if you're not familiar with Tay-Sachs, if you're not familiar with gout, those are things that should go on your flashcards, not so much what gene it is, because I intentionally put that in parentheses for just sometimes to remind myself when I'm talking about it. And sometimes in things that are in parentheses will be outside parentheses in other slides. So that's not a blanket exemption if it's in parentheses. Oh, don't worry about it. That just means right there in that sentence, it's for explanation. Peroxisomes are vesicles filled with oxidative enzymes. So here's our peroxisomes, and these are just little cartoons. That's not necessarily what they look like. They detoxify compounds and fatty acids. The process of oxidation is using oxygen to remove a hydrogen from a substrate. And peroxisomes do that. They have that in their name. They are oxisomes. They detoxify drugs and alcohol. And the chemical equation, the reason I put this on here is so we can see how using oxygen to remove hydrogen works. So here we have hydrogen peroxide, and we're bringing in some this R1 just as a substrate that has hydrogen attached to it, and we're going to use this hydrogen peroxide to oxidize this hydrogen. We're going to take it, and we have 2 hydrogens here, 2 hydrogens here, so 4 hydrogens, 2 oxygens, and here we're going to bring that hydrogen peroxide in and make water. So 4 hydrogens, 2 oxygens, it's balanced. We still have the substrate, but we took that acidity off of it, and so it helps us detoxify those compounds. Catalase is an enzyme that, and you'll get a clean copy of these slides too without all my writing on it, but catalase is an enzyme that catalyzes a peroxidative reaction. So the peroxides, kind of like what I showed you here, but just a straight up peroxide. We have catalase enzymes that when the peroxide is generated, and we'll see when we look at inflammation that we generate some of these radicals, that enzyme can just straight break it down into water and oxygen. So that also happens in peroxisomes. Peroxisomes are used in the synthesis of some lipids, some specialized phospholipids for nerve cell myelination. Next, we'll look at mitochondria. The mighty mitochondria are used for cellular respiration and energy production. They have a double membrane with embedded enzymes. They contain their own DNA coding for enzymes needed for metabolism. So mitochondrial DNA is actually a little bit different than regular nuclear DNA. They kind of have their own DNA, and they can actually replicate themselves, and that's one of the things that happens if you do a lot of aerobic conditioning. Your mitochondria will actually replicate themselves to help increase the efficiency of your metabolism. The inner membrane contains the enzymes of oxidative phosphorylation, which is responsible for ATP production. We'll look at that a little bit later. They conduct cellular respiration. Cellular respiration includes the citric acid cycle, and this is one of those places where the citric acid cycle can be called a lot of different things depending on the textbook and when it was published. The citric acid cycle is also known as the Krebs cycle or the TCA or tricarboxylic acid cycle. All 3 of those refer to the same thing. Within the mitochondria, we also have the electron transport chain. It produces water, and it's the main place where ATP is formed. Versus glycolysis, which does not occur in the mitochondria. Glycolysis occurs out here in the cytoplasm, outside the mitochondria. And that also produces energy, but not nearly as much as the mitochondria. The cytoskeleton provides cell structure, movement, and internal transport. Its main mode of taking care of those things is called mechanotransduction. Mechanotransduction is a cellular process that translates mechanical stimuli into biochemical signals. So something happens to that cell, it gets stretched, it gets pushed, something mechanically happens to it, and that converts into a biochemical signal. And the cytoskeleton helps to modulate that. It allows the cell to adapt to its surroundings. But if there's a dysfunction, it may cause or be associated with a outcome of cardiovascular disease, a loss of hearing. If there's a problem with the cytoskeleton, that may cause or be correlated with a alteration in an organ system that loses its capacity, or not loses necessarily, but may alter its capacity to carry out this normal mechanotransduction as it normally would. Within the cytoskeleton are microtubules and microfilaments, and they are there for structural support, intracellular transport, and cell movement. And we'll talk about those individually when they come up later. Objective 2, review the structure and function of cell membranes. The plasma or cell membrane encloses the cell. It separates the extracellular fluid from the intracellular fluid. It allows transport between the extracellular fluid and intracellular fluid, though, but it's selectively permeable. Not everything can get through. We'll look a lot at how that's regulated. It also provides for recognition and communication through receptors. The membrane structure is amphipathic, or also known as amphiphilic, and it's a bilayer. It's primarily phospholipids, but there's also some cholesterol and some sugar-linked molecules. The sugar-linked molecules are called glycolipids and glycoproteins, and that glyco, let me get my pen here again, that glyco is the sugar telling us it's a sugar attached to a lipid or attached to a protein, and together those make the glycocalyx. The actual phospholipids have a hydrophilic charged phosphate head here and two fatty acid hydrophobic tails here. And so you can see each of these is a phospholipid with its hydrophilic phosphate head, hydrophobic fatty acid tails, and they arrange themselves into this bilayer. They spontaneously organize into the bilayer. They're not forced there, they're not bonded together, but it is energetically favorable because the fatty acids are hydrophobic. They like to be together like a drop of oil would be, and so they face each other and they spontaneously arrange this way with the hydrophobic fatty acid tails sandwiched between the hydrophilic phosphate heads. So they're attached to one another but not bonded. It allows for fluidity and easy damage repair. Something gets poked through the membrane, you poke a needle through the membrane and pull it back out, then they just move away and then come back together. You don't pop it, it's not like a balloon. They just get out of the way and then they come back together. It's impermeable to water-based molecules and permeable to lipid-based molecules, which will be very important when we start talking about signaling. Water itself does cross the membrane through aquaporin protein channels. Membrane proteins provide function to the cell. The phospholipid bilayer up here, if you look at this membrane, this is all phospholipid bilayer here, giving the cell structure, the membrane, but all of these are proteins. These green squiggly things are proteins, and they give function. The type and number of the proteins depend on the cell location and what its function is. The proteins are formed by ribosomes and finished at the Golgi, like we talked about a few slides ago, and then they move by vesicular transport to the membrane where they're inserted. They can be embedded either interior or exterior, and here we see one that's exterior kind of anchored on there and interior kind of anchored on there. Or they can be transmembrane, which means they span the whole membrane on both sides, from interior to exterior. They're highly folded with hydrophilic substance surface amino acids and hydrophobic interior ones. So as they form those coils, as you look at through the membrane there, the inside of those coils would be hydrophobic, and then the outside out here facing the extracellular fluid would be hydrophilic, so that they can interact appropriately with wherever they are. Proper folding is required for adequate function. If they're misfolded, that increases the risk of disease, which we'll talk a lot about when we talk about Alzheimer's and different protein folding and the problems that come when those proteins don't fold correctly. Proteostasis is the process of maintaining that homeostasis of the protein folding. So proteins are formed at the ribosomes. Here, ribosomal, we've got the translation at the ribosomes, and it has to go through not just translation, but it has to go through the proper folding and I'm losing the word here. Proteins are chaperone proteins here, and they're going to help with the folding. And then, but we also have to have a way to, if they're misfolded and if they're dysfunctional, to get rid of them. And we do that using the proteases. They will degrade those, and we'll talk more about that process in future chapters. But I kind of want to remind us that it's not just it goes through the ribosome and everything's happy. It also has to go through all these folding processes, and that doesn't always work right. And so we have to have mechanisms for correcting that if it's incorrect. And that's part of that protein homeostasis process. Protein functions, they can be used for recognitions as receptors. So for cell signaling to activate, if we've got receptors, a ligand can come in and generate a signal. We'll look a lot at that. Pores and channels, they can transport ions, amino acids, whatever they're designed for, they can transport them through the membrane. Enzymes are a big one. Enzymes cause products and substrates. They can take in a substrate and form a product, catalyze a reaction. A lot of proteins are enzymes. Sodium potassium ATPase it's actually a pump, active transport pump. I should have separated those out because enzyme pumps and active transport pumps and enzymes are a little bit different things, but they probably because they had it the same on here. But we'll talk about sodium potassium ATPase. I did have it separated out. I was just ahead of myself. Enzyme catalysts, those are going to cause the conversion of a substrate into a product. For example, metabolic chemical reactions, and we'll look at those pathways. Cell surface markers. When we talk about the immune system, we'll really talk about cell surface markers and what makes our cells unique from somebody else's cells. And those are what are attached to their proteins. Glycoproteins, immune system recognition, we'll talk a lot about those later. Cell adhesion molecules, those you can see link. Those proteins join cells together, so that just like their name says, they adhere cells. So they can attach to the cytoskeleton and contribute to cell shape. Cells surround themselves with a secreted extracellular matrix. That matrix is formed by fibrous proteins embedded in a complex carbohydrate-rich gel. And you can see that this one's kind of less organized over here, so you can see more components in this extracellular matrix. But here, this is part of the interstitial matrix. Here outside, this is showing you an epithelial layer and then a basement membrane underlying that, and then the blood vessel lying right under. But the blood vessel also has membrane, and so this matrix here, this extracellular matrix here, kind of is between those different types of tissues, the blood tissue and the epithelial tissue, and it holds everything together. It's not that our tissues are just slipping and sliding around next to each other. Everything's kind of adhered by this matrix. In the matrix are things like collagen, and collagen can form cable-like fibers and sheets, and that provides tensile strength to the tissues and resistance to longitudinal stress, for example, in a joint capsule. So our tissues don't just fall apart like paper. They have some stability to them from the collagen. Also, elastin is another fibrous protein. It's rubber-like, it bounces back, so if you stretch a tissue, then it can recoil like the lungs. So elastin in places where there needs to be a lot of stretch and recoil, like in the aorta, you're going to find a lot of elastin. So the extracellular matrix forms that basement membrane underlying the epithelial tissue, and that's that. Here they've got it, they just have it kind of circled and then they call it basement membrane over here. Under that epithelial tissue attaches connective tissue cells like bones and cartilage within the tissue. Allows for diffusion pathways for nutrients, waste, and other water-soluble traffic. So we'll see that when we look at capillaries, we'll see that fluids will come out of the capillaries and feed into this interstitial matrix, and then fluids can go back into the capillaries as well, and we'll look at that in chapter 3. Contributes to cell mechanical support, so the cells have some stability with that basement membrane underlying them. Proliferation, we'll see that the interstitial matrix, the extracellular matrix, is going to play a huge role in cancer. Because when we talk about cancer, in a lot of ways is wound repair that's gone awry, and wound repair is tightly regulated by this matrix. And so we'll look at that a lot more when we look at cancer. Tissue regeneration, scaffolding, that comes into that wound repair as well. So we'll come back to this matrix later in future chapters when we look at those kind of processes. Also maintenance of tissue microenvironment. So what is the nutrient capacity around that tissue, and what is the immune status, all those kinds of things. Cell junctions join cells and allow communications. They're tight junctions which form barriers to leakage. They're going to form a tight seal. You can see a tight junction here. And then they help maintain the cell polarity too. What is the apex and what is the basal side? So the cell knows which way is facing the lumen and which way is facing the basement membrane, because those will have different kind of directional components to them. Desmosomes are epithelial bands. Here's a belt desmosome and a spot desmosome. They're strong. They brace the tissues for structural stability. So strong mechanical attachments like rivets and these strong belts that are going to hold those cells together. And you can see that up here, a belt desmosome that's holding those cells together almost like a packing strip. And then gap junctions are clusters of tunnels that allow substances to go through. They allow chemical communication and tissue coordination. In this picture, they look kind of like those tight junctions, but this is supposed to show that there's a channel between there where substances can go through, and so communication can occur. Really important in the heart for communication in heart cells. Each cell in that gap junction contributes a connexin hemichannel. So a connexin means they're coming together, and a hemichannel is half a channel. So each one comes together on each side of the membrane and has this half channel, and together, here's one cell and here's another cell, they form this channel coming together to form that pore. Objective 3, describe the modes of cellular communication. Cells must communicate to maintain homeostasis and regulate function, growth, and development. Communication occurs via receptor binding and subsequent activation of cell signaling. We're going to look at those processes. It may be contact dependent if the cells have membrane-membrane contact, like through a gap junction. So the gap junction may coordinate activities involving adjacent cells. Or ligands may travel a small distance, like neurotransmitters, or a greater distance, for example, hormones. When we look at communication, there's this whole framework of physiology that we're going to look at. We start with a ligand, and a ligand is going to be our first messenger, and it's going to come up to a cell, and it's going to do one of two things. If it's a peptide ligand, it's going to bind to a receptor, and that receptor is going to cause a signal that will either stimulate some sort of product release, or it may cause a chemical, electrical, or mechanical alteration, or it may signal DNA to go through that process of transcription and translation to form a gene product. So the ligand will stimulate the receptor to do those things. There are some ligands that may actually inhibit that, but we'll look at the stimulatory process first. But if that ligand is a lipid, it might bypass that receptor process, go straight to the DNA, and stimulate that transcription and translation process there. There are feedback loops at every stage, and this process, this framework of physiology, is mainly what we're going to look at throughout all of the chapters. What signals are the cells getting? Why are they responding the way they are? That's physiology. And then what happens if that response is altered? That's pathophysiology. So this is the process that we go through in looking at each of our organ systems and seeing what should it be doing and what went wrong and why did that go wrong. And so we follow this process as we go through. These kind of diagrams where we've got stimulations and inhibitions are called causal loop diagrams or systems models, and we'll be working with these on the Friday here in orientation. Tuesday we meet together for physiology, and then we will also meet again on Friday to talk more about these types of drawing these models. When we look at physiology, the biggest trouble students have in the graduate level pathophysiology is moving from undergraduate to graduate thinking. Because in undergraduate thinking, in most cases, you were taught, here's the cardiovascular system in its little box of cardiovascular system things. Okay, now let's put that box away and let's look at the respiratory system and the respiratory system things, but you didn't necessarily learn to integrate them and integrate with the kidney. And so we kind of have to change our way of thinking. Memorizing won't work because we're going to integrate all the systems. We can't separate them into little boxes. So using these systems models will help us do that. And we'll talk about that during orientation. The process of chemical communication, cell communication, is called signal transduction. We have to transduce the signal from one form, a chemical form, to either another chemical, an electrical, or a mechanical, and that's a transduction. We're changing forms. Cellular communication may occur through the interaction of a ligand with the receptor. So here we have a ligand, and here is its receptor. And when those interact, that's going to generate a signal. A ligand is a chemical messenger, might be a peptide hormone, might be a neurotransmitter. It's going to bind with a receptor. So the receptors recognize and bind the ligands to initiate that cellular response. The response may be direct, like opening and closing a specific ion channel, or it may activate a second messenger system, and that will cause a cascade that's going to amplify that signal. So here we have that relay and amplification. When we look at receptor types for these ligands, they can be channel linked. Channel linked are receptors that open and close in direct response to a signal. The ligand will bind to a specific membrane receptor closely associated with that channel and change the electrical status of the membrane. For example, sodium and potassium movement. So if we look at this one, here we have this extracellular chemical messenger. It's going to bind to this channel, and when it binds, the sodium and the potassium are going to change their flow. So sodium comes in. Here comes sodium in, and that it doesn't show, maybe I colored over it, doesn't show the potassium going out, but it's going to cause the movement during an action potential where sodium influx causes depolarization, potassium efflux causes repolarization. Those can use different channels. The sodium potassium ATPase can change the influx and efflux to get them back to normal. There's going to be a bunch of different channels, and I'll actually show you a picture of all the channels that are involved just in action potentials. But they can be activated by these chemical messengers. Normally for depolarization and repolarization, we're dealing with voltage regulated channels, and voltage regulated channels would be activated by a difference in the membrane potential. So that's something a little different than the chemical ones they're showing here. There can be mechanical deformation of a channel. They don't show that one on this picture. For example, stretching. There can be catalytic enzymes activated by ligand binding, and we'll look at some of those. And then G protein linked. G protein linked receptors mediate the interconversion of GTP and GDP to activate a second messenger, and that's going to be through a G protein intermediary, and we'll look at those too. So if we look at G protein linked receptors, with a G protein linked receptor, and these are one of the most common, and that's why we look at them, we have this first messenger ligand, and the first messenger ligand is going to bind to its receptor. It activates the G protein intermediary here when the ligand binds, stimulates the adenylyl cyclase, that's an enzyme, conversion of ATP into cyclic AMP. Cyclic AMP is a second messenger. So the first messenger was the binding ligand, but it can't get in. This receptor is not a channel. The ligand is going to bind to the receptor here, but it's not actually going to get into the cell. It does not get into the cell. Instead, it sets up these dominoes that are going to trickle down, and it's going to activate this G protein intermediary, which activates the adenylyl cyclase enzyme, which converts ATP into cyclic AMP, which is a second messenger. That's going to go and it's going to activate a protein cascade. I'm getting ahead of myself. We also, in addition to the ATP, we could also do this with GTP and convert GTP into cyclic GMP. In that case, it wouldn't be adenylyl cyclase, it would be guanylyl cyclase, because it would be, but it's going to be the same effect. Then, once we get the second messenger, whether it's cyclic AMP or cyclic GMP, it's going to amplify the signal by binding with these protein kinases, and that's going to change their shape. And then when we change their shape, they're going to have an altered function. So that altered protein function generates then a cellular response. For example, cyclic AMP is a second messenger that's involved in the signaling catabolism of glycogen, breaking down glycogen to release glucose when epinephrine and glucagon stimulate the receptor. So if here's our receptor, if our first messenger here is glucagon or epi, they're going to come in, and that's going to generate the second messenger cascade, this G protein cyclic AMP cascade. That's going to cause glycogen to be broken down into glucose. I'm still learning to write with my pen. Again, it's been a few months. But that would be inhibited by insulin, because if insulin is there, we don't want more glucose if insulin's there. We don't want our glucose levels to go up. So insulin would actually inhibit that reaction. Cyclic GMP, one of the things that cyclic GMP mediated is by the binding of atrial natriuretic peptide. Then that would activate the cyclic GMP that activates a protein kinase to induce smooth muscle or blood vessel wall relaxation. And we'll look more at the effects of ANP here in a little bit. The binding of that first messenger to a G protein can also activate a different type of second messenger pathway. This case, we're looking at the inositol phospholipid pathway. In this case, when the first messenger binds to the receptor, it activates this G protein intermediary that's going to, in this case, activate the phospholipase C. And what phospholipase C is, it's an enzyme that's going to take one of these phospholipids from the membrane. It's just going to pop a phospholipid out of the membrane, and it's going to enzymatically convert it into diacylglycerol and inositol triphosphate. So those second messengers. They will then generate a second messenger cascade that's going to really result in the release of intracellular calcium from storage. Calcium interacts with the protein calmodulin, and then that, just like the kinases in the previous slide, that's going to alter the protein shape and cause a cellular response. For both of these systems, whether it was the cyclic AMP system or this inositol triphosphate system, the impact is that the first messenger doesn't get into the cell. The first messenger binds to the receptor, and that activates this second messenger response. So the first messenger comes to the gate, and then the gatekeeper activates the response. And the response can always be the same as far as the second messenger, but depending on the type of cell it is, that's going to change what the cellular response is. We don't need a lot of receptors. We don't need a lot of second messengers because the output will depend on the type of cell. It's like if you're going to a restaurant and you're going to place an order, that's the first message. But the waiter here or waitress here is your receptor. They're receiving your order. The kitchen is the same. Where this all happens is the same. The process is the same. But what comes out depends on what you ordered here. And you know, you're not going to go to a restaurant that specializes in, you know, pasta to order a burger usually. You know, you have different kinds of places for different kinds of things, and same with cells. If you're going to make insulin, it's going to be at the pancreatic beta cells, so they're specialized. But the second messenger system within it might be the same as a different cell that does something completely different. So in this case, with the calmodulin, it can mobilize the calcium to initiate blood vessel smooth muscle cell contraction in response to angiotensin II receptor activation. Objective 4. Describe the major steps of cellular respiration to produce ATP. Metabolism is all the chemical tasks of maintaining cellular functions. It includes an anabolism, which is using simple molecules to build complex ones. That uses energy, and catabolism, which is breaking complex molecules into simpler ones, which is releasing energy. The way I remember these two is I always think of a cat, and a cat breaking things down and releasing all this energy. And so that just reminds me which one's anabolism and which one's catabolism. Metabolic pathways take the simple molecules and break them down further to extract the energy from their chemical bonds. When we look at something like glucose, so glucose has 6 carbons and it's going to have oxygens and hydrogens attached, and I won't draw all those. But we're not interested in the carbon. We actually want to get rid of that. We breathe it out. We want this bond energy. That's what our cells need, is that energy in that bond. So we put compounds into the chemical pathways, the metabolic pathways, so we can break them down and extract that energy from their chemical bonds, because it's the energy we want, not the atoms. The atoms we breathe out as carbon dioxide or we release as water. We want the energy that held them together. Proteins, which are complex molecules, are broken down into amino acids in pathways. Fats, which are complex molecules, are broken down into fatty acids in pathways. And starch and glycogen, which are polysaccharides, complex molecules, are broken down into simple sugars in pathways. So we have pathways to break all these components down to get their energy out. Other pathways then would take these simple molecules and build them back up into things, and that would be anabolic pathways then, building them back up into those things that the body needs. Metabolic pathways are managed by protein catalysts called enzymes. Enzymes are proteins with a specific affinity for a particular substrate, and each pathway will have many, many enzymes. The enzymes convert the substrates into the products, and they can also convert them the other way, from the products into the substrate. Usually an enzyme can go either way, but the direction it goes is determined by the concentration. If we have a lot of substrate, then it's going to keep putting it into products, especially if those products then are taken away, and that maintains that gradient in the direction that's favorable to go from substrate to product then. Depending on the relative substrate product concentration gradients and the chemical energy state, that's going to determine how those enzymes work. For example, hexokinase is an enzyme that attaches a phosphate atom to glucose to prime it for the glycolysis pathway. So simple sugar is glucose. Before glucose goes into glycolysis, it gets a phosphate slapped onto it. We eventually get that phosphate back as ATP. It gets put onto an ADP. But the phosphate gets put on so that the glucose stays in the cell. So it can't get back out with the phosphate. So it kind of traps it in there. Pathways used to transfer energy from the chemical bonds in order to form ATP are some of our main metabolic pathways. So all of these on here are ones that are feeding into this process of making ATP. So all of our citric acid cycle and glycolysis and all the pathways that feed into that process. So glycolysis, citric acid cycle, electron transport chain, those are our main ones. ATP is a chemical energy used to drive cell processes. That's the cell currency. We need ATP to get it to do its work. So all these pathways are geared towards making ATP. Must be continuously replenished by metabolism. The equation for the breakdown of sugar, so if we're taking a simple sugar and we're taking it through this whole process all the way down through glycolysis and the citric acid cycle and the electron transport chain, we start with glucose and we add oxygen and we get out carbon dioxide, water, and energy in the form of ATP. And we'll look at each of these processes a little bit. Energy is transferred chemically from one molecule to another, but some energy is also lost as heat. But what we want is in the bonds of this glucose, we want to extract the energy and store it as ATP. We get rid of the water, we get rid of the carbon dioxide. We don't want those atoms. We want this energy, that ATP. That's what we want from that sugar. To make ATP, we have to have that energy to attach a third phosphate onto an ADP. ADP is a diphosphate. It has two phosphates. ATP has a third phosphate. And that adenosine with three phosphates, this is a really high energy bond. And we really want to store energy in that bond because we can use it later. That's energy in the bank. So we take that energy that we got from the breakdown of sugar and we're going to use it to attach this phosphate onto this ADP to make ATP. ATP is not stored in large amounts. We can only store a little bit of it. The body stores chemical energy primarily in the bonds of fats with some in glycogen, because then as fats we can put them into, break them down into fatty acids and send them into the citric acid cycle through the acetyl coenzyme stage, and that process is called beta oxidation. ATP is used for anabolism of organic molecules, muscle contraction, active transport, primarily of sodium and potassium to maintain electrical and chemical membrane gradients. Any cell need that's not supplied by random kinetic motion requires ATP. So we have to keep making huge amounts of ATP in order to keep the metabolism moving forward. Some examples of catabolism are extracellular digestion, where enzymes break apart polymer macromolecules to release small monomers, amino acids, simple sugars for absorption into cells. We'll look at that when we look at the gastrointestinal system, where all those enzymes come in, all those enzymes of the stomach and the pancreatic enzymes and the amylases and lipases and all those. We'll look at those in the GI system. But that's an example of catabolism, breaking big molecules into small ones. Glycolysis in the cytoplasm is an example of catabolism. In glycolysis, here's our glycolysis process. We start with glucose, which has 6 sugars. That's why it's got the C6 here for 6 carbons. And we're going to break it into 2 pyruvate molecules, each of which has 3 carbons. And from that, we get some ATP. We had to use 2 ATP, but we're going to get 2 back. And we generate these 2 NADHs, and we'll see where those come in a little bit. So we're breaking apart that glucose into these smaller molecules. Most of the pictures I use will be from your textbook. However, in some of this more review stuff, I do pull from a Creative Commons undergraduate A&P book. So that's freely available online if you want to go look at the OpenStax A&P book. And that's where I get some of these pictures if I use some of the undergrad pictures for things that we need to kind of go back and review a little bit. So you can always go online and find that. That's a free book, free PDF. Adenosine triphosphate. In making ATP, it's combined processes of anabolism and catabolism. So we are breaking some things down, but we're also building. So it's kind of a combination. The ATP is formed by the oxidation of glucose to carbon dioxide and the reduction of NAD to NADH. So you can see here that when we go into this transformation of pyruvate stage, that we're going to release some carbon dioxide. So we're breaking apart because the formula for glucose is C6H12O6. And the carbon and the oxygen are going to come out as carbon dioxide. Now we need a little bit more oxygen that comes from water, and we'll talk about that here in a second. And then the hydrogens will go onto the NADHs, and we'll take those out down to the mitochondria here in a little bit. So the NADH will release that hydrogen down here to, let me find where the step is. Here it is. So the NADH, when we get down, and we'll talk about the mitochondria in a little bit, it's going to release those hydrogens into the electron transport chain, and then through that process we'll make ATP, and I have a better picture for that here in a little bit. But what I want to point out here is that this NADH is going to release the hydrogen to the inhaled oxygen here. We'll bring in this inhaled oxygen, and the oxygen is reduced to form water. This is actually a critical step because up here I said we brought in some water to make this oxygen. Here we have to inhale oxygen to make that water. The reason this is such a critical step, and the reason I emphasize it so much, is that sometimes there's a big misconception. One of the biggest misconceptions I see in cellular respiration is that people think we need to inhale oxygen to form carbon dioxide, and that is absolutely not true. You're going to make carbon dioxide no matter what. Even if you are suffocating and cannot get any air, you are still going to make carbon dioxide, even if you're not bringing in one speck of oxygen, because your carbon dioxide comes from sugar, and your muscles still have to break down sugar. Where your inhaled oxygen comes in is making water at the end of the process. By that time, if you're not breathing, it's too late because you've already made so much carbon dioxide here. This is where your inhaled oxygen comes in here. You've got to have that inhaled oxygen to make water, not to make carbon dioxide. You make carbon dioxide out of glucose, and it seems like I'm harping on this minor thing. It's really, really important when we get to the respiratory system. And even when we talk about acid-base later today, that we understand what the role of carbon dioxide is and where carbon dioxide comes from in the body. Carbon dioxide comes from the breakdown of glucose. It does not come from inhaling oxygen. So that's critical. After glycolysis, the pyruvate then, and I kind of mentioned this on one of the previous slides as an example of something different, that pyruvate will enter the mitochondria. That releases, as it does that, it releases a carbon dioxide and forms an NADH, and it's going to happen twice because there's two pyruvates per glucose. The remaining two carbons, because remember this is a 3-carbon, 1 carbon left here is carbon dioxide. We have 2 carbons left in the acetyl group. That's going to bind to the coenzyme A. The coenzyme A will shuttle that acetyl group into the Krebs cycle, also known as the citric acid cycle or tricarboxylic acid cycle. And then the cycle is going to go around twice and once per acetyl, and it's going to generate 2 ATP. It will generate 6 NADHs, 2 FADH2s, and 4 carbon dioxides. The reason for that is, remember, we're starting out with glucose, C6H12O6. O6. What we want from the glucose is the bonds that are holding it together, not the atoms. We're getting rid of the atoms. We're getting rid of the carbon and oxygen. We're getting rid of the hydrogen. What we want is the energy. Water is contributed to provide the extra oxygen here at this stage to make the carbon dioxide. And the reason I say extra oxygen is because we have 6 carbons and 6 oxygens in glucose, but we make 6 CO2s, so that's 12 oxygen. So we need a little bit extra oxygen. It comes from water, not from inhaled oxygen. And I really harp on that, and it gets annoying because I harp on it so much, and I ask you questions about it so much because it's so important. It's so important to understand where that carbon dioxide comes from, and it's a really common misconception. So the oxygen intake comes in down here to make water. Carbon dioxide will still be made. You're making carbon dioxide here. You're making carbon dioxide here. You're making carbon dioxide here. Well, this is just a summary up here. And you've already made carbon dioxide here even if you were not breathing. So you're already changing your acid-base balance. By the time you bring in oxygen and to breathe in oxygen, it's all the way down here. Oxidative phosphorylation is where we're going to make the majority of our ATP. It occurs in the electron transport chain. We mentioned in the previous few slides that in glycolysis and in the Krebs cycle, citric acid cycle, we attached hydrogen to this NAD and the FADH2. What those things are, they're B vitamins. Nicotinamide adenine dinucleotide and flavin adenine dinucleotide, NAD and FAD, they're hydrogen shuttles. They're going to take this hydrogen off of glycolysis, off of the Krebs cycle, and they're going to take it here to the inner mitochondrial membrane where these cytochrome proteins are. If there's a B vitamin deficiency, that's going to disrupt the metabolism because then you can't get the hydrogens to the cytochromes if you're missing the NAD or the FAD. NAD and H and FADH2 provide the hydrogen to the inner mitochondrial membrane cytochrome proteins. They pump it into the intermembrane space here. So all these hydrogens are getting pumped in here and they're building up an accumulation of hydrogen. They're building up a concentration gradient. That concentration gradient then is used like a waterfall for that hydrogen to go down through the ATP synthase enzyme here. And as it goes down through this concentration gradient, using the energy of the concentration gradient, that energy gets used to slap another phosphate onto ADP and make it ATP. So it's going to have that high energy. So that's how we make the majority of our ATP, is through this ATP synthase, using almost like a hydraulic engine, using that hydrogen power to move down its concentration gradient. And that energy is then trapped in that third bond of ATP. So hydrogen diffuses back through ATP synthase. Let me make sure I'm not missing anything. To form ATP, this is where the inhaled oxygen enters. Okay, so here's where this inhaled oxygen enters. It's going to take this hydrogen and oxygen and form water. So the inhaled oxygen doesn't come in till the very end of the process. We need it, absolutely we need it, so that we can neutralize that hydrogen, but we need it at the end. The system will back up without inhaled oxygen, but carbon dioxide production is not going to stop. If you're suffocating and you have no oxygen coming in, you're still going to make carbon dioxide because you're making it from the breakdown of sugar. So that the alterations in carbon dioxide are still going to happen. So what if we don't have enough oxygen? Then we have to go through substrate level phosphorylation. That occurs in the absence of oxygen. If oxygen is available, we're going to go through oxidative phosphorylation. So in that case, what we would do, if oxygen was available, we'd go through that pyruvate system we just saw. We'd go to acetyl coenzyme A, that would go into the citric acid cycle, that would transmit our NADHs to the electron transport chain, and we'd bring in oxygen and we'd combine it with the hydrogen that's coming out of the electron transport chain, and we'd make water. In that process, we take the NADH and we pick up a hydrogen off of pyruvate here, and then that hydrogen on the NADH will go down to the electron transport chain. But if there's no oxygen, that process is going to back up and we're not going to be able to go this route, so we're going to have to go one of the fermentation pathways. And there are two fermentation pathways. There's the fermentation to make lactic acid and the fermentation to make alcohol. In both of the fermentation pathways, instead of taking a hydrogen off of pyruvate, we're going to add one on. So we take a hydrogen off of NADH and we add it on to the pyruvate to make a molecule. They're a little bit different because this one has one less carbon, so it's got a carbon dioxide coming off. But in either case, we're adding a hydrogen on. Anaerobic glycolysis occurs when the pyruvate plus that hydrogen then is attached and becomes lactic acid. It is reversible when the oxygen supplies are replenished. So when oxygen comes back, it can go back this direction, go back to pyruvate, and then go down through this process here when the oxygen comes back. Increased tissue lactic acid therefore indicates tissue hypoxia, because we're not going to build up this lactic acid unless we don't have enough oxygen to take care of this NADH. Results in less ATP production versus oxidative phosphorylation. The best way to make ATP is go through that electron transport chain. That gets us the best bang for the buck as far as. Objective 5, compare and contrast the types of membrane transport. Cells use membrane transport to regulate the intake, so nutrients, fluids, chemical messengers that are getting in, output, what kind of metabolic and lysosomal digestion products are they putting out, ion movements, sodium and potassium movements, all of that is regulated by membrane transport. It may be passive. If there's passive transport, that means we don't have to put in any energy, no external energy. There's still energy, but it's following the laws of physics. Random kinetic motion, entropy, those are what are driving passive transport. Active transport requires ATP input. So if we're going to transport something actively, we have to give it a push, and we use ATP to give it that push. Solute passage through a membrane depends on size, solubility, electrical charge, concentration gradient. Lipid-soluble molecules can go right through the membrane. So the oxygen, carbon dioxide, nitrogen, urea, those can go right on through. Small uncharged molecules can go right on through the membrane. Now in this case it has water. Water actually is charged and it does have special aquaporin channels, so that's a little misleading, but anything that's small and uncharged can go right through that membrane. Small charged water-soluble molecules are going to have to pass through pores or channels. So those would be your ions. So they have a charge. So they can't go just right through. They're being rejected here by the membrane because they're going to have a channel. Electrolyte concentration in the intracellular fluid versus the extracellular fluid. So this is our ECF and this is our ICF. Varies, and it is going to impact the transport rates, and it's important in signal transduction in nerve and muscles, and we're going to talk a lot more about that in chapter 3. Pores and channels may be gated and triggered by ligand binding or voltage change. We'll talk a little bit here in just a little bit about gated and triggered ligand voltage change channels. We'll talk about action potentials. Electrolytes are electrically charged and they dissociate into ions. So anything that has these positives and negatives are ions. They carry an electrical charge. Cations are positive charge and anions are a negative charge. So your cations are things like sodium and potassium, and anions would be things like chloride. Ions tend to migrate toward an area of opposite charge. So not only do they move by their concentration, but charge also has an impact. If they're anions, they're going to move towards a positive charge because anions are negative. If they're cations, they're positive, so they're going to move towards a negative charge. So that kind of complicates the picture sometimes. The concentration may vary intracellular fluid and extracellular fluid. For example, there's a high potassium in the intracellular fluid versus a high sodium in the extracellular fluid. Ion movements are going to alter the voltage, and that's important for electrical transmission. If a positive ion is moving, it takes that positive charge with it. You can't separate the ion from its charge. So it's going to change the charge of wherever it's leaving from and going to. The way we measure charge is by milliequivalents per liter. That's the number of electrical charges per liter. And it's not based on the size, it's based on the valence. So sodium, potassium have one positive charge, so they're considered monovalent, whereas some ions like calcium and magnesium are divalent because they have two positive charges. That's going to also cause a difference in their concentration electrical. Diffusion is the movement down a concentration gradient from greater to lesser concentration. So sodium, if sodium is high outside the cell, its movement into the cell is by diffusion because that's the way it wants to go chemically for equilibrium. So movement is due to random kinetic energy because that promotes equilibrium. That's what it wants to do by laws of physics. So distribution moves towards equilibrium across a permeable membrane. Small and or lipid soluble components would move more rapidly. Sodium's small, it's not lipid soluble, but there are channels for it. Diffusion is faster with a larger concentration difference. Simple diffusion is across a membrane. Facilitated uses a protein pore or a channel. Movement may be influenced by the channel charge. So some channels have a charge inside and that can impact it. For example, calcium-linked channels may slow the rate of sodium and potassium movement because calcium is positive and so are sodium and potassium, and like charges repel. Osmosis is the movement of water down the water concentration gradient. Water moves from high water, low solutes, to low water, high solutes. So in this example, there's more sodium outside the cell, and if that was the only thing impacting it, which it's not, but in this example, let's say that was the only thing impacting it, water is going to be drawn towards the sodium because there's more water in this example inside the cell than outside, so it's going to be drawn out. Now, in reality, there's a lot of potassium inside the cell. Osmotic movement's related to osmotic concentration. It's not related to the particle size or the weight. So it doesn't matter how big the ion is or how big the particle is. If it's big protein, it is more related to the osmotic concentration, not the actual size of the particle. Osmolarity is the osmoles of solute per liter of solvent. We also use the term osmolality, which is the osmoles of solute per kilogram of solvent. Usually in biological systems, if we're talking about fluids in the body, we use osmolality because it's easier to measure kilograms than it is to measure liters inside of a body. So osmolality is more commonly used. An osmole is a unit of osmotic concentration. An osmole is how much osmotic pull that substance has. What kind of pull is it drawing that water with? Osmole generates osmotic pressure. 1 mole of sodium chloride in solvent is 2 osmoles because sodium chloride is going to dissociate into a sodium ion and a chloride ion. So it is 1 mole of sodium chloride, but 2 osmoles because you have 2 ions that dissociate. Biological fluids are usually milliosmol measurements. If we had pure water, osmolality would equal osmolarity because 1 liter of water is a kilogram. In the body, the osmolality will be more than osmolarity in cells because there are other things in that solvent, but it's really very minor difference numerically. Usually it's a matter just of convention that if I was talking about fluids in the body, I'd say osmolality, And if I was talking about like a bucket of fluid like urine, I could say osmolarity because I could measure that in liters, whereas the body fluid is harder to measure in liters because you'd have to know how much fat there was and all that kind of stuff. But it's really a minor distinction. Body osmolality is about 280 to 294 milliosmoles per kilogram. The extracellular fluid and intracellular fluid concentrations equalize over time due to ongoing osmosis. So if one changes osmolarity, if the extracellular fluid changes osmolarity, the intracellular fluid will reach equilibrium with that over time. The osmotic pressure of a solution is measured from its opposition. The osmotic pressure is how much hydrostatic pressure is required to oppose the osmotic movement of water. So we kind of measure it in the negative. So an osmotic pressure is how much hydrostatic pressure we'd have to apply to prevent the osmotic movement. Osmotic pressure occurs in capillaries and the nephron glomerulus. It's influenced by the concentration gradient, the solubility, the molecule size, the membrane thickness. Effective osmolality is called tonicity. So what it's actually doing in the body is called its tonicity. Sustained osmotic activity that depends on the concentration of solutes remaining on one side of a permeable membrane is the tonicity, and it's lost if equilibrium is reached. For example, plasma proteins retain effective osmolality within capillaries because they don't cross the membrane versus urea, which does, and we'll look at some examples of all of this. Plasma proteins have a negative charge, so they're going to alter the distribution of other anions, for example chloride, in response to cation movement. So that we can't discount the impact of plasma proteins. Since they have a negative charge, they're going to impact everything else too. Filtration is the movement of water and solutes across a membrane due to a pushing force. So in this picture we have hydrostatic pressure. Hydrostatic pressure is the weight of water pushing down on this column of fluid, and we're pushing out the fluid here by filtration. So across this is our membrane, and we're pushing water out through that membrane by filtration because that's the hydrostatic pressure pushing on it. So this is filtration. Hydrostatic pressure is the mechanical force of the water pushing against that membrane. It occurs when the water pressure on one side of the membrane is greater than the other. Blood hydrostatic pressure causes filtration from the capillaries. It's opposed here by this oncotic pressure. And the proteins here are acting like a magnet. That's why it's got the magnet. The proteins, since proteins can't cross the membrane, I mean, I don't really like this picture because it does show actually more proteins over here. Although actually this is, I suppose, water. But I don't know why it's pointing to this yellow thing on proteins. It's kind of a confusing picture. But if there were proteins over here, which let's say there are because of this magnet, that's going to pull the fluid this direction back towards those proteins. And that's going to, we call that the oncotic pressure or the osmotic pressure. The draw of those plasma proteins will cause it, and that's called reabsorption. And we do use a P, not a B. Not reabsorption, absorption. It just doesn't, the P makes it easier to phonetic, or it's more of a language thing as far as how we pronounce it. Diffusion and osmosis are passive transport. They rely only on the energy provided by random kinetic motion. Active transport, on the other hand, is protein-mediated transport that requires the input of energy. So for active transport, we have to put in ATP. For active transport, we must have receptors capable of binding and recognizing the substance to be transported. For example, the sodium-potassium ATPase pump antitransporter. It's an antiporter, which means if the sodium's going out this direction, the potassium's going to come in the other direction. They go in opposite, so they're called an antiporter. It's going to take 3 sodiums out, use the ATP to provide the energy, put that phosphate, it's kind of putting that third phosphate in this little spot, kind of like a key to provide the energy, and that's going to power the pump. So the sodium comes out, the potassium goes in, potassium goes this way, sodium goes that way. Here we have potassium going in, the phosphate goes off, and it's going to need to find an electron transport chain or something to attach it back to another ADP to make ATP again. So we've used up that energy that was binding that phosphate to the ATP in order to transmit our molecules. So we provided energy. So 3 sodium out versus 2 in maintains the net membrane charge with negative inside cell negative relative to the outside. This pump actually uses the majority of cell ATP production, especially in nerves, muscles, and the kidney. Other examples of active transport: calcium concentration regulation in skeletal and cardiac muscle. It has to be pumped into the sarcoplasmic reticulum and sequestered there so it's not causing muscle contraction. Glucose and amino acids symport with sodium in the intestines and kidneys. Glucose and in the intestine and kidney cells, when sodium is absorbed, glucose comes along with it, so it's called symport. But we set up a sodium concentration gradient in order to draw it down and pull that glucose with. ATP is required to maintain the sodium concentration then necessary to support this transport. So it sets up the sodium concentration gradient, and then sodium, as it goes down its concentration gradient, pulls glucose with it to be reabsorbed. If there's a loss of active transportability, that's pathophysiological. For example, if there's a disruption of nerve transmission, muscle contraction, kidney function, any of those things can occur if there's a loss of ATP production. And you'll lose ATP production if you lose oxygen because you can't maintain your electron transport system efficiently then. Large molecules such as proteins pass into and out of the cell by endocytosis and exocytosis. Endocytosis is a section of plasma membrane invaginating and forming an endocytotic vesicle. So it's going to pull the molecule in to the cell. Cellular uptake of receptor ligand complexes, and it kind of shows that in this one. We've got some invagination of the receptor complexes. It'll show it on, I think I have another picture, a better picture on the next slide too. So it can pull in extracellular components, bacteria, viruses, cellular debris. It's activated by the ligands binding to those membrane components, and then once they bind, it causes that the cytoplasm to invaginate that and change the vesicles, and then they're called coated pits, and then they invaginate and pull that stuff into the cell with it. Pinocytosis is ingestion of the extracellular fluid and the contents. It's called phagocytosis if it's a large amount. So once that stuff's pulled in, then it might also bind with a vesicle and fuse with a lysosome if that's something that needs to be digested, not just to get rid of it, but if it needs to break it down into components that the cell can use. And all that is regulated by what is bound and what is triggered and all the cellular signaling mechanisms. Exocytosis, so here there's kind of a better picture of endocytosis too. So the particle gets invaginated and then it can bind with a lysosome and get digested if needed. Exocytosis is when the contents leave then. So we've got something that is made in the Golgi or finished in the Golgi, made in the ribosomes, finished in the Golgi, and then put into a vesicle, and it leaves through the cell surface. It's the main mechanism by which molecules are discharged at the cell surface into the extracellular matrix. So for example, the release of insulin from pancreatic beta cells. The plasma membrane is internalized in endocytosis. So if we only did endocytosis and we were making all these vesicles, we'd lose, every time we made a vesicle, we'd lose some plasma membrane. It would get smaller and smaller and smaller. But then when we do exocytosis, we give some plasma membrane back. The membrane doesn't leave the cell, just the contents. The membrane fuses with the cell membrane and then it becomes part of it, and so then it would flatten out and it makes the membrane bigger. So we lose some membrane by endocytosis, but we gain it back by exocytosis, so the cell stays roughly the same size. plasma membrane regularly undergoes invagination to form endosomes. Within endosomes, smaller vesicles bud internally to form these multivesicular bodies. So here we have a multivesicular body with all these vesicles inside of it that have been formed by endocytosis. It allows the cell to sort components and compartmentalize them so it can keep fluids and nucleic acids and proteins in different microvesicles so that it can be organized. This is a very active area of research and we don't know a whole lot about it yet, but I bring it up because some of these new areas are going to be very significant moving forward as you go out into practice, and they learn more about this and they learn more about how these cells are really organizing all of this stuff. And so this is a big player in some types of immune things that they're looking at. Microvesicles are then secreted. Here you can see the exosome release here and the products leaving. When they bind with the plasma membrane, depending on what signal is given, what needs to be excreted. But it allows for very precisely differentiated secretion. It's not just this whole vesicle that's being released, it's individual components, individual microvesicles that can then fuse to the membrane and release their contents. Very organized. Not a whole lot known about it, pretty new. It's an active area of research found in all studied body fluids, everything they've looked at, blood, urine, amniotic fluid, cerebrospinal fluid, tissues. So it's a big component that not a lot's known about yet. They know it's important in cell signaling, immune system activation, facilitation of cancer metastasis. So a lot of research into this area. Objective 6, describe the mechanisms involved in the generation and transmission of action potentials. Excitable cells at rest are electrically polarized with the inside more negatively charged than the outside. The resting membrane potential is -70 to -85 millivolts in the interior, depending on the type of cell, and it's due to differences in the ion and protein composition of the intracellular fluid versus the extracellular fluid. Because proteins carry a negative charge, the inside of the cell tends to be more negative. Sodium is higher in the extracellular fluid with potassium higher in the intracellular fluid. This is maintained by the sodium-potassium ATPase pump, this gradient. At rest, the potassium diffuses outward more easily than the sodium diffuses inward, and that also helps maintain the gradient. A change in membrane potential allows the transmission of information. A stimulus leads to an increase in sodium influx, and we call that depolarization. Depolarization by 15 to 20 millivolts reaches threshold. If threshold is reached, a rapid sodium influx occurs next through the voltage regulated channel. So once we reach threshold, we get a massive sodium influx. The sodium, not all of it like I've shown in this picture, it's just a little bit, but enough goes in that it changes the polarity of the membrane. It's an all or nothing response. Once we reach threshold, we've tipped the scales and it's going to occur. It's like standing on a diving board. If you lean and you lean and you lean, you're going to reach threshold and you're going to go in the water. You're going to reach a point where you cannot go back. Depolarization brings the interior charge towards zero, and it may actually flip that charge to positive. Threshold occurs at the point where inward and outward currents are equal and opposite but unbalanced. When we look at this picture, what this is showing us, this is a action potential diagram, and this is the threshold here, membrane threshold, and this is the regular membrane potential where it is at rest. So if it's at -90 at rest, it has to get to a little bit like -65 here before it reaches threshold. Once it reaches threshold, it's going to generate this action potential, all or none. It's going to go all the way if it reaches threshold. If it doesn't reach threshold, if it only gets up maybe this far, it can still go back. But once it reaches threshold, it's going all the way, all or none response. Any further depolarization at threshold will activate enough voltage regulated sodium channels. It's going to tip the system to an action potential. Any deviation of ions, sodium, potassium, calcium, from normal may change where these threshold or membrane values are, and are going to change how this cell behaves. And we'll look at some examples of that. When the interior becomes depolarized, so we saw that now this sodium's inside the cell. So the sodium's inside the cell. So we're at the depolarization stage up here. When that cell becomes depolarized, voltage-gated potassium channels are activated. Potassium then exits the cell in response to the electrical and concentration gradients. And the electrical gradient, because it wants to move away from like charge, it got positive in here. So that means potassium's like, I'm out of here. I'm going out here. And then that's repolarization here. As the potassium leaves, it's taking its positive charges with it, and the inside becomes more negative then. So the cell interior becomes more negative again. That's called repolarization. So you can see that we started here at -90. As sodium came in, then it brought its positive charges with it, and it got more positive. Then as potassium goes out, it took its positive charges out and it got more negative. So we repolarized. Ions, though, were on the wrong side. Now the potassium's on the outside, sodium's on the inside. So the cell's in a refractory period. So the charge is right, but the ions are wrong. So we have the absolute refractory period where no response to stimuli occurs, and a relative refractory period where there's a response only to a stronger than normal stimulus. So sodium-potassium ATPase is going to reset those ion gradients by moving 3 sodiums out for every 2 potassiums that come in, and it's a major source of ATP use. So it's going to reset that gradient, but it's going to require a lot of ATP. If we look at this, I like this picture better. This is actually from the 7th edition of McCants. And they didn't have it in the 8th edition, I don't know why. But here in the resting state, you can see you've got your sodiums on the outside, and then as you start to go up with depolarization, the sodiums start to come inside. They're pulling their positive charges with. And then as you go down for repolarization, your potassiums start to leave, and they're pulling their positives with them on this side. But then your sodium-potassium pump will have to reset that gradient. So it's going to pull 3 sodiums out for every 2 potassiums it pulls in. So it's going to reset that gradient. It seems like it would get unequal because it's pulling 3 sodiums out for 2 potassiums, but we have leak channels for potassiums. We have more leak channels for potassium. So you can see this arrow's a little bigger because the potassium leak channels are more common than the sodium leak channels. So it has to have that 3:2 ratio to balance. I mentioned before that it's a more complicated picture than just sodium comes in and potassium comes out. We use a simplified model, but the cell membrane ion channel complex is much more complex. This is actually a cardiac myocyte, and this is a picture that I drew that hangs on my wall in my office because it's a very complex picture, and it's not just the sodium coming in. I mean, you can see sodium coming in here through the voltage gated channels, but look at all these potassium channels that we're dealing with, and they don't even all go out. We've got some that go in. It's a very complex picture as far as what's actually happening. Over here we have a sodium calcium channel, and so it's a lot bigger picture than if you learned in undergrad, probably what I just showed you, that you have your action potential, sodium in, okay, sodium in, depolarization, potassium out, repolarization. In reality, it's a much more complex picture than that. Do not memorize this picture. You don't have to know that. The reason I show it to you is so that you can kind of appreciate that what you learned in undergrad is maybe not quite the whole picture. And so sometimes when people ask me questions, I'll say, well, what do you tell me what you know? Because I need to ascertain where you're actually at. and where you're coming from, because everybody will have a different experience as far as what they took and what they learned and what they remember. So some of the stuff that we go through in here is going to be different in some ways than what you've learned before, because now we're going to the next step and we're taking our simplified model and we're making it more realistic. And even this picture doesn't even include everything. This is still a simplified picture for what's going on. Don't memorize that. I just want you to appreciate that the picture is more complex than what you may have learned before. Objective 7, review the cell cycle. Cell cycle is the process of preparing for and carrying out cell division. It includes both mitosis and cytokinesis, which is somatic or body cell division, and meiosis, which is gamete production of eggs and sperm. We'll talk about just the mitosis part in this little bit, and then we'll talk about meiosis later, not in this chapter. Interphase is cells in preparation for cell division. In this picture, this cell is in interphase, but you can also see, if you're familiar with mitosis, we have a cell in prophase, metaphase in the middle, anaphase, separating, and then telophase, 2 cells. So this cell is in interphase, but this slide also shows mitosis here. Cells carry out normal daily activities when they're in interphase. Cells replicate their chromosomes and organelles that they need for cell division. G1 is the time span between the end of mitosis and the start of DNA replication. It's really variable. It depends on the tissue type, and it's short in tissues that replicate frequency, like coverings and linings, skin, intestines. It's long, essentially unending, in tissues that do not replicate, so nerves, muscles, lens of the eye. And there's actually also a G0 phase that some of those cells can just kind of go into and stay in if they're never going to divide. The S phase comes after the G1 phase, and the S phase is DNA replication. In DNA replication, the sister chromatids duplicate. There's no change in chromosome number, but the sister chromatids go from diploid unduplicated to diploid duplicated. We have 23 duplicated chromosomes, so 46 chromosomes. But when we're coming out of mitosis is here, and we're coming into G1, all of these chromosomes are unduplicated. So here's a chromosome from mom. Let's say this is mom's number 1, this is dad's number 1, and chromosome number 1 of 23. And we're going into the S phase, and we still have mom's number 1 and we have dad's number 1. And they're what are called diploid because we have 2 chromosome number 1s, but they're unduplicated. During the S phase, we're going to take those unduplicated chromosomes and we're going to make a matching sister chromatid. So we're still diploid because we have mom's chromosome number 1 and dad's chromosome number 1. But now they've gone from unduplicated to duplicated because now each of them has a sister chromatid. G2 is the time span between DNA replication and the start of mitosis. This is where RNA and protein synthesis occur in preparation for cell division. And then we enter mitosis, which is nuclear and cytoplasmic division. That's where our prophase, metaphase, anaphase, telophase, and cytokinesis are going to occur. Here the sister chromatids separate. There's still no change in chromosome number, but we go from diploid duplicated to diploid unduplicated. So back here, we made these duplicated chromosomes. We still have those here. So we're diploid. We still have mom's chromosome number 1, dad's chromosome number 1. But in mitosis, the sister chromatids are going to separate and go into different daughter cells. And now we're still going to have mom's 1 and dad's 1. But the other cell will also have mom's 1 and dad's 1 because the sister chromatids separated. This sister chromatid went here. This sister chromatid went here. So we go from diploid duplicated in mitosis to diploid unduplicated. That's going to be important when we start looking at genetic abnormalities, that cell division process, and also when we start looking at meiosis in the genetics chapters. I emphasize that diploid duplicated to diploid unduplicated and back again because it will come up again. So this is a case where if you're not familiar with those just off the top of your tongue, make a flashcard because it's important to learn this terminology now because it will come up again. Organ and tissue growth and repair depend on the balance of the rates of cell division, cell survival, and cell death. The cell cycle control systems involve a series of biochemical switches that either activate or inhibit cell events. These switches are cell-dependent kinases. They're protein kinases that regulate phosphorylation of intracellular proteins to start and regulate the cell cycle. They must join with cyclins to form a cyclin CDK complex for activity. What happens is there are these checkpoints, these gates that the cell has to pass through. And in G1, this cell-dependent kinase has to match up with this cyclin, this G1 cyclin, in order to pass this check mark. Here, there's this S cyclin that this cell-dependent kinase has to bind with. And here, to enter mitosis, it needs an M cyclin. So there are these checkpoints that it has to pass through in order for correct progression through the cell cycle. If something happens to these cyclins, this is where a big place where cancer can occur, because there's an alteration in these cyclins, these cell-dependent kinases, and that alteration can mess up the whole progress through the cell cycle and make it unorganized. And so there's all these checkpoints and all these cytokines and all these complexes that have to be formed at the correct time in the correct order in order for the cell cycle process to occur, so we're not just haphazardly causing cell division. Activation then would be inhibited by binding of a cell-dependent kinase inhibitor protein, so a CKI. These work in concert with other regulatory proteins. An example of regulatory proteins are mitogens. Mitogens are chemicals that stimulate mitosis, so they're going to trigger that cell-dependent kinase activity. They stimulate the mitogen-activated protein, and that's a component of the RAS system. So this is the RAS system we'll look at here. And this is just an example. There are many different types of regulatory pathways. The RAS system is a GDP system. It's a G protein activated system. And it's going to activate the GTPase, and then when the GTPase is activated, then we have active RAS. The active RAS is going to phosphorylate other proteins, and here's some of those other proteins. We don't really want to worry too much about those details of those proteins being phosphorylated. But what we do want to look at is the MYC protein, myelocytomatosis viral oncogene protein down here. Because if this RAS system is activated abnormally, it can abnormally activate the MYC protein. And that is a protein that has a gene coding for a transcription factor. If that transcription factor is always on, if it gets mutated so it's always on, the cell cycle is always going to keep progressing. And you're going to keep pumping out these cells and growth and cancer because the normal regulation isn't working, because the cell has become, this gene has become mutated and it's running out of control. Something I want to make a note of here, RAS stands for rat sarcoma. It's actually a common cell signaling in our cells, but it was found in rat sarcoma cells, and that's why it's called that. Same with this myelocytomatosis viral oncogene. That's where it was found, but it's in our cells too. And so sometimes we get these crazy names and it's not fun and they don't make sense, but by the time we realize that they're in all cells, that name has already been established, and it would be confusing to change it to cell cycle biologists. So sometimes we get crazy genes and proteins that don't seem to make any sense, but they're important. Cytokines stimulate protein synthesis and inhibit degradation. Any alteration in cytokines, and cytokines are any of these signaling proteins, any of these pathways, Any alteration can increase or decrease cell growth depending on the specific cytokine involved, and we're going to look at a lot of those in immunity, and we'll look at a lot of them when we look at cancer. So I'm kind of introing it here, but we will come back to this kind of process. It does impact the cell cycle. Tissue specialization involves distinct patterns of gene stimulation, growth, and division. Organelle distribution, membrane protein types and distributions, all of those things are going to impact how a tissue develops out of a cell growth. If it develops along the coverings and linings path, it's going to form epithelial tissue, and then it's going to be specialized for absorption and protection. Connective tissue has abundant extracellular matrix of ground substance and fibers. Muscles are contractile cells. Neural tissues receive and rapidly transmit electrical signals. Stem cells are where these all start. And stem cells retain their multipotency, which is the ability to become any kind of terminally differentiated cell. So it can become a muscle, it can become a bone. A stem cell can be anything. And so the stem cells are where we start with that process of growth. Next, we're going to move into Chapter 3, the cellular environment, and it follows right on. A lot of the things we talked about in Chapter 1, we're going to do in Chapter 3. We will do Chapter 2, but we put it with the immune system. It fits better there. So objectives for Chapter 3, review the distribution of total body water and the role of osmosis in fluid balance, identify the mechanisms that cause edema, define isotonic, hypertonic, and hypotonic water and solute alterations, identify the terminology and major consequences associated with abnormal levels of sodium, potassium, calcium, phosphate, and magnesium, review acid-base homeostasis and associated buffering systems, and compare and contrast the causes and compensations of metabolic and respiratory acidosis, metabolic and respiratory alkalosis, and describe what is meant by the anion gap and explain the significance of an abnormal anion gap in metabolic acidosis. So we start with Objective 1, review the distribution of total body water and the role of osmosis in fluid balance. And if you have not taken a break yet, pause this right now and go take a walk, because we're at 1 hour and 45 minutes. 44 minutes and 54 seconds. So I really meant what I said, that I break these down into objectives. You should only go through one objective at a time and then take a break, because it may seem productive, but if you haven't taken a break yet, you've been zoning out. So take a break. So we're going to talk a little bit about body water and osmosis now. Homeostasis relies on proper balance of acid-base ratio, electrolytes, and water. Balance is maintained by integration of renal, endocrine, and neural function. Electrolytes regulate excitable cell function and osmotic balance. And pH alterations disrupt enzyme function. And we're going to look at each of these statements, and we're going to see what happens with these. Water moves freely through cells and tissues. It's distributed by osmotic and hydrostatic forces, and we'll look at those in detail. Total body water is about 50 to 60% of the kilogram weight of an adult body, about 42 liters. 2/3 of the body water is intracellular and about 1/3 is extracellular. The excuse me, extracellular fluid is mainly interstitial fluid and intravascular fluid or blood plasma. The percent water decreases with increased percent fat. Now, with increased body size, that doesn't mean there's less water, there's more, but the percentage decreases because the fat doesn't hold water. So the percentage, it's not an absolute decrease, it's a percentage decrease. Infants store less fat, so their water's higher, can be up to 70 to 80% of their body. That means that fluid loss is especially dangerous in infants because it can very quickly lead to dehydration, because any loss in the extracellular fluid is going to pull intracellular fluid to balance it, because that fluid is always going to go to equilibrium. With aging, that percent can vary as regulation is disrupted by decreased organ function. And we'll look at that more when we look at the kidneys, but it depends on how the function is. There's too much being lost, too much water being lost, too much being retained. By organ dysfunction can go different directions. So it's going to depend on the actual dysfunction. Equilibrium of osmolality. Osmosis balances water between the intracellular and extracellular fluids. Diffusion through the plasma membrane occurs at aquaporin water channel proteins. The osmotic regulation of the extracellular fluids primarily based on sodium concentration. Remember that most of the extracellular fluid ions are sodium. So there's more sodium out here than there is intracellularly. On the other hand, the intracellular fluid has more potassium, so potassium is going to have more of an impact on the intracellular fluid. Extracellular fluid's more likely to change because that's where you're going to lose blood, you're going to lose fluid in the urine, those kinds of things. And then the intracellular fluid is going to mirror that change. So if the extracellular fluid becomes dehydrated or overhydrated, osmosis will do the same thing to the intracellular fluid over time. In extracellular fluid, fluid is balanced between the plasma and the interstitial fluid. Fluid moves from the capillaries to the interstitial space by filtration. So as fluid moves out of the blood into the interstitial fluid, that occurs by filtration. It's due primarily to this capillary hydrostatic pressure, also called the blood pressure or blood hydrostatic pressure. Fluid then moves back from the interstitial space back into the blood because we don't want all the fluid leaving the blood. We go into shock. So it comes back by reabsorption. So reabsorption pulls it back in. That's mainly due to these proteins. We saw that in chapter 1, that these proteins are generating that colloid osmotic or oncotic pressure, and that's pulling like a magnet, pulling that fluid back in as those plasma proteins go up. Net filtration pressure is the balance between the blood hydrostatic pressure pushing outward by filtration and the blood colloid osmotic pressure pulling inward by reabsorption. Net filtration pressure, NFP, is the net filtration pressure. That's going to move fluid out of the blood at the arterial end of the capillary and into the blood at the venous end. Any fluid not reabsorbed, any fluid that stays out here and does not get reabsorbed, is going to go into the lymph and should be taken back to the heart. So the net filtration pressure is a balance between our blood hydrostatic pressure and our blood colloid osmotic pressure. Understand the relationship between these. Don't just memorize the equation, because to understand the kidney later on, you have to understand, and part of the cardiovascular system, you have to understand this relationship. Hydrostatic pressure in a vessel is the volume pressure here. So it's the fluid, not these proteins, but the fluid. How much fluid pressure is pushing that filtration outward of the fluid. Now there is hydrostatic pressure out here opposing that, but not very much usually. So just a little. This is kind of dominating. On the arterial end, it's the blood pressure, or the blood hydrostatic pressure, that's pushing the fluid out by filtration. Colloid osmotic pressure draws that fluid back in. As more of this volume is lost by filtration, these proteins become more concentrated, and as they become more concentrated, they pull that fluid that's out here that was filtered, they pull that fluid back into the blood at the venous end of the capillary. So that's going to pull the water back in. So as fluid leaves the arterial end of the capillary, the water hydrostatic pressure decreases. Since the proteins are too big to filter, they become more concentrated. So as this water leaves, the proteins are becoming more concentrated here, and eventually they're going to reach a concentration capable of pulling that water back in by the colloid osmotic pressure. So the net filtration pressure here is going to be positive, and here it's going to be negative. That's not very clear. Negative. So if it's negative, that means we're not filtering, we're doing the opposite of filtering, which is we're reabsorbing. So we can look at that a different way. So over here we start, and this is a systems diagram, and like I said, we'll talk about these and we'll do more of these throughout the semester because it's important to understand this integration. So we start with a high blood volume, and our high blood volume is going to stimulate our blood hydrostatic pressure. Our blood hydrostatic pressure here is what's pushing us out. So we're going to push out from the capillary and we're going to stimulate filtration. I can't write it. I'll misspell things, but go with what I say because I can't write and I don't like to stop. It's going to take a long time if I stop every time I need to do that. All right, as the fluid gets filtered, that's going to inhibit the volume. The volume is going to go down because the fluid went this way. It went out. It went this way. And so the volume is going to go down. But as the volume goes down, that stimulates the proteins. We don't make more proteins, but they become more concentrated. So I'll put stimulates protein concentration. So that is BCOP, blood colloid osmotic pressure. As the proteins get more concentrated, that blood colloid osmotic pressure is going to inhibit filtration. So the filtration, though, as we pull the blood colloid osmotic pressure, that water that was filtered is going to be pulled back in by those proteins. So it's going to be pulled in by those proteins, and that's going to then stimulate an increase in blood volume. Here. Anything that's not picked up by the increase in volume is going to go to the lymph. With these diagrams, it seems like, I mean, first time you look at one of these, you're like, I'm not doing that. I'm just going to memorize the words. But trust me, if you try to memorize the words, this is chapter 3, you're going to be of an 1800-page book, you're going to be completely overwhelmed. Once you learn to do these diagrams, it really makes it much more clear. And we'll talk about that more when we get to campus, and we'll do lots of practice ones together. But drawing these out, and it's really important to draw your own, not just to memorize somebody else's, because you have to go through the mental process of making your own mental model of how this works. Otherwise, you can't jump in the middle and take a test question where I come at it and go backwards, or I come sideways, or I come in from the middle and expect you to know what's going on. So it's really important to draw out these for yourself instead of trying to memorize a bunch of words, because you'll get completely overwhelmed. But don't worry, we'll practice and we'll do a lot of them together. If we go the pathway to the lymph, what's going to happen is that we can get edema. So objective 2, identify the mechanisms that cause edema. Edema is an excessive accumulation of fluid within the interstitial spaces. It's usually due to unequal fluid distribution, not necessarily total body water excess. So it may result in intravascular dehydration, which seems really counterintuitive. You know, if you've got this much fluid out in the feet, it seems like, well, then the cells should be full of fluid too, but that's not how it works, because there's some problem in the lymph that's not draining this fluid, and so it may actually, the cells may become dehydrated, which seems really counterintuitive with edema. It may result from increased blood capillary hydrostatic pressure, decreased plasma protein concentration, and we'll look at each of these mechanisms, capillary membrane damage resulting in altered permeability, lymphatic obstruction. May be localized, it may be generalized, depending on where the problem with the lymph is. So increased capillary hydrostatic pressure. Remember that capillary hydrostatic pressure, or blood hydrostatic pressure, is the pressure that's stimulating filtration here. So we're stimulating filtration by our capillary hydrostatic pressure, also known as blood hydrostatic pressure. Remember, capillary hydrostatic pressure is the same thing as blood hydrostatic pressure. The increased fluid volume in the capillaries, because there's so much, it's pushing, that volume is pushing that fluid out by filtration into the interstitial space. So we've got a lot of filtration here, high filtration, pushing into the interstitial spaces. This may result from venous obstruction. So if we've got a clot down here, then that fluid can't go there, so it stays here and it gets pushed out this way. Same deal for restrictive clothing. That's going to resist the outflow because there's a blockage here. It's not allowing it to drain, so it can build up, especially if it's in the legs, it can build up back down in the feet. Prolonged standing, because gravity can pull that fluid down to those capillaries of the feet and then cause that increase in blood pressure and cause that increase in filtration. Sodium or water retention, which can be due to the right heart failing, and so it backs up. If the right heart normally pushes blood to the lungs. If it's backing up, then it's backing up into the systemic system. If there's renal failure and it's not getting cleared, if there's liver cirrhosis, and liver can mess up a whole bunch of stuff. So that's going to mess up your proteins too, and we'll talk about proteins here in a minute. That can cause volume overload and increase the venous pressure. That's going to increase the filtration. The volume of interstitial fluid exceeds the capacity to go to the lymph, so it can't all go to the lymph. That was weird. So it builds up out here and you get edema. Increased capillary permeability. So let's say maybe the pressure's normal, but we've got holes in the, there's too much permeability. If there's too much permeability in the capillary from inflammation, if the cells were damaged, if there was burns, neoplastic disease, allergic reaction, infection, anything that increases that permeability, which allergic reaction and inflammation are big, or infection are big ones, because capillaries increase their permeability to help the immune system deliver the products it needs. So that's going to allow even more fluid, because it's not going to have to go through through the membrane. It's just going to leak out through these pores, and lots of fluid's going to leak out through these pores. Lots and lots of fluid. So we're going to lose all this fluid out through these pores. Problem also is that, remember, the protein should stay inside of here. Protein, 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 protein. They should stay in here. They should not get out. But if these pores are big enough, some of the protein can get out, and then we've got an even bigger problem, because if the proteins get out here, we know what proteins do. They draw fluid, so then they're going to draw all this fluid out with them by osmosis, and we're going to lose even more fluid. So that's going to draw fluid out and to the interstitial fluid by osmosis, and can make the problem even more severe, because now all that fluid's out there. It's not going to go back because it doesn't have the osmotic pressure to go back, because the protein's out here now. So now if it can't go all off to the lymph, then it's just going to stay here and build up edema. Decreased plasma oncotic pressure. If we have normally in our blood vessel here, we normally have all these proteins. And as we get on this side, then the fluid leaves over here, and we draw back in from these proteins from that blood colloid osmotic pressure. But if the proteins aren't being made because there's a liver problem or something like that, then we don't generate that pressure and we don't pull that fluid back in, and we get edema. We have to have those proteins in the blood in order to pull that fluid back in. So if there's liver disease, protein malnutrition, kidney damage, so we're losing proteins in the kidney, we're losing blood, we're losing drainage, seepage through wounds and burns, any of those that loses the proteins, we lose that draw to pull those back in, and we have to have those proteins to pull that water back in. So this is a huge problem with if there's any kind of liver disease or kidney disease. If we don't make those proteins to pull that fluid back in, then that fluid's going to go out in the tissues, and not only does that cause edema, but that can cause circulatory collapse too if we're not pulling that fluid back in. So that decreases the osmotic pull into the plasma, so it prevents the reabsorption of fluid from the interstitial fluid, and we get that edema. Lymphatic obstruction. If we have this process of this filtration, that's normally reabsorbed, and some of it goes to the lymph, But if these lymph vessels are blocked, then that can't be pulled back into the lymph. So maybe there's a tumor, maybe there's an infection blocking them, or maybe there was an excision. They took some of the lymph nodes out to check for a biopsy, and now there's no way for it to drain. So after the lymph nodes have been removed in cancer, then it can't drain correctly. So in that case, they're not reabsorbed. They can accumulate. And if they accumulate, then we get the same thing. We get edema because it's not draining. So that can cause lymph. Objective 3, define isotonic, hypertonic, and hypotonic water and solute alterations. Water follows electrolytes. We saw that when we looked at osmosis. The water will go to where there's more electrolytes. So water and sodium are especially linked due to its high concentration in the extracellular fluid. Sodium concentration is regulated by aldosterone in the adrenal gland and atrial natriuretic peptide in the heart. We'll look at those. Water is regulated by antidiuretic hormone in response to changes in blood osmolality. So we'll look at antidiuretic hormone as well. It's released from the posterior pituitary gland. 90% of our extracellular fluid cations are sodium. So sodium is outside the cell. Its charge is balanced because the solution is neutral. And so it's balanced by anions, chloride, and bicarbonate. When we look at a cell and we say, oh, the cell's a negative interior, that's just right at the membrane. It's not the whole fluid all through the cell. It's just right at the membrane. So it can be, there are proteins through the cell, so it can, I guess, be distributed through the cell. But we mainly look just at the protein for that change in charge. Chloride's usually transported passively and follows sodium. Chloride and bicarbonate concentrations usually vary inversely, and they can compensate for one another to some degree. So they're both anions, and so if chloride goes down, bicarbonate might go up a little bit and balance each other that way. These ions, the sodium and chloride and bicarbonate, are going to regulate that extracellular fluid osmotic balance. Maintenance of the extracellular fluid sodium concentration is crucial to the formation of action potentials. So we saw the action potentials in chapter 1, and we know that we have to maintain those concentration gradients in order to generate those action potentials in the excitable cells. Sodium is used in acid-base buffering through the actions of sodium bicarbonate and sodium disodium phosphate, so we have to maintain those concentrations. Blood sodium concentration is regulated within the kidney by fine-tuned control of reabsorption. Any sodium that is excess will be lost in the urine. So let's look at how this process occurs. If there's any kind of decrease in blood pressure, renal blood flow, or blood sodium, that's going to stimulate the release of renin from the nephron juxtaglomerular apparatus. What renin or renin, some of the pronunciations may differ very depending on how you were taught it and different regional pronunciations, so sometimes I go back and forth with those two. Renin is an enzyme that catalyzes the conversion of angiotensinogen, which is made in the liver, into angiotensin 1, and that's a vasoconstrictor. So renin will, if blood pressure goes down, if blood volume goes down, renin will be activated to convert that angiotensinogen into angiotensin 1. Angiotensin 1 is converted into angiotensin 2. That's a more potent vasoconstrictor, and that's converted by angiotensin converting enzyme primarily in the pulmonary blood vessels. Angiotensin 2 is a vasoconstrictor. It's going to vasoconstrict blood vessels to increase the blood pressure. It also stimulates aldosterone release from the adrenal gland, and aldosterone helps to retain sodium, and then water is going to follow that sodium as it's retained, and that will increase the blood volume. Increased blood pressure and sodium at the juxtaglomerular apparatus then will work as that water retention occurs. That's going to, and vasoconstriction occurs. That's going to inhibit those by negative feedback, and then that release will discontinue. Aldosterone is a steroid release from the adrenal cortex. It stimulates increased sodium reabsorption from the tubular filtrate into the blood at the nephron distal convoluted tubule. I think there's an error in the book. I don't know if it's this chapter or somewhere it says proximal convoluted tubule, but aldosterone is distal convoluted tubule. Water absorption follows the sodium, increasing water retention. The thing to remember about aldosterone is aldosterone increases sodium retention and water retention, but in the kidney it's going to retain sodium, so it's going to pull sodium, retain that, but it loses potassium in antiport for that. So the potassium then can go into the urine and be lost. Atrial natriuretic peptide released from the heart, brain natriuretic peptide, which is also released from the heart, and urodilantin. That's one of those examples of they found something and named it and then they realized where it was coming from. And urodilantin, which comes from the kidney. These are all natriuretic peptides, and what they do are they're hormones that are hormones released in response to tissue stretch. So when the blood volume and pressure are too high, that's going to stretch the tissues of the heart, and as it stretches those by that mechanical change, that's going to cause the release of atrial natriuretic peptide and brain natriuretic peptide, and those are going to go then out to the kidney. They increase sodium loss. Aldosterone increase retention, but ANP is natriuretic, which means sodium into the urine, and it's going to lose sodium. So it's going to induce vasodilation of the afferent arterioles. That's going to increase the glomerular filtration rate and increase sodium filtration and loss in the urine then. Natriuretic peptides oppose the outcomes of aldosterone and angiotensin II. They work to decrease the blood volume and decrease. Successful water balance requires monitoring of the fluid pressure by arterial baroreceptors and hypothalamic osmoreceptors. Increased plasma osmolarity and/or reduced blood volume or pressure stimulates antidiuretic hormone or vasopressin. Those are the same hormone. Vasopressin and antidiuretic hormone are the same hormone released from the posterior pituitary. This is a case where they thought there were two different hormones doing two different things because there are two different receptors, and then they found out later they were the same hormone. And so it has two names, but it's the same hormone. It's released from the posterior pituitary gland. And what antidiuretic hormone does is it goes out and increases the permeability of the distal nephron and collecting duct to water, so it's going to help retain water at the kidneys. And so that will increase the blood pressure then. Reduces blood osmolality by dilution because it's retaining the water. But when you retain the water in the blood, that means you take it out of the filtrate, and so the urine osmolality is actually going to increase, or osmolarity, because once it's urine, then you can measure its volume, and so it's okay to say osmolarity then. Release is inhibited by negative feedback. As the blood volume and pressure go up, then you're going to lose the stimulation to pump out that antidiuretic. So if we look at this whole picture, so when we're looking at tonicity, we're looking at blood volume and blood pressure. If it's too high, that's going to cause the heart to stretch. We're going to release atrial natriuretic peptide, and we're going to lose sodium and water at the kidney. If blood volume and pressure are too low, we're going to do two main things. We're going to release vasopressin and antidiuretic hormone from the posterior pituitary. Those are going to cause vasoconstriction. Vasopressin means blood vessel constriction. An antidiuretic hormone is going to retain antidiuretic, so we're going to oppose diuresis, so we're going to retain water. The other mechanism is we're going to activate the RAS system, and the RAS system stands for renin, angiotensin, aldosterone system. And so we're going to activate that renin process where we convert angiotensinogen into angiotensin 1 into angiotensin 2, which is a vasoconstrictor, so it will act in concert with the vasopressin. And we're also going to increase aldosterone to activate sodium and water retention. So we've got a lot of this type of system, especially for the volume pressure falling low, is called a redundant system because there's more than one way to solve the problem. More than one thing working in concert. We'll come back to the RAS system over and over and over, so I kind of want to introduce it here to kind of refresh your memory on this endocrine system, because it's going to become, it'll be so important. We're going to come back to it lots of times, lots and lots of times on RAS. So when we talk about tonicity, an isotonic solution has the same osmotic solute versus water balance as the plasma. So in an isotonic solution, if you put a cell into an isotonic solution, we have the same, let's say it's 290 milliosmoles. An isotonic solution would be the same inside the cell as outside the cell. So there's still going to be water and solutes going back and forth, but they're at equilibrium. So any changes in electrolyte concentration are accompanied by proportional changes in total body water in an isotonic. So let's say we lose fluid through hemorrhage, we're losing blood, so it's still isotonic. Our volume may go down, but our tonicity is going to stay the same. We could gain isotonic solutions if there's an intravenous isotonic replacement, but that may long-term alter the osmotic balance even if it's isotonic, because if it's dextrose or saline, that's going to impact the concentration gradient, and we'll look at that more when we look at sodium, the impact of sodium. Due to the osmotic relationship between sodium and water, a sodium imbalance is going to lead to a water imbalance. So that's going to come into play if we have one of those osmotic changes due to the IV, but it's also going to come into play when we look at our hypertonic and hypotonic solutions. Any change in tonicity can change the electrolyte concentration relative to the water volume. So if we have a hypertonic solution, a hypertonic solution is when the extracellular fluid osmolarity rises above the intracellular fluid osmolarity. So let's say the intracellular fluid is still at 290 milliosmoles, but our extracellular fluid goes up to 295. Now that's a hypertonic solution because the extracellular fluid has a higher tonicity, has a higher osmolality than the intracellular fluid. It can happen because sodium increases, or maybe there's a decrease in water, so it becomes less concentrated. But either way, what's going to happen is, in order to balance that by osmosis, since we have less solutes inside and more solutes outside, water is going to go in this direction. That can dehydrate the cell. Hypotonicity would be the opposite. So in this case, let's say our cell is still at 290 milliosmoles, but in this case, the extracellular fluid is at 285. 285 milliosmoles. In that case, we have fewer solutes out here, and our extracellular osmolality is lower than our intracellular. And that might be because our sodium's decreased, or we have too much water. There's a water toxicity going on. And in that case, water is going to get pulled into the cells because the cell is more concentrated of solutes, so the water is going to move into the cells. Unfortunately, that can swell up the cells. Both of these alter the concentration gradients because water is moving, it's going to dilute either the extracellular fluid or the intracellular fluid, and that's going to alter the concentration gradient. If you alter the concentration gradient, you can alter your membrane gradients as far as your action potentials and your enzyme functions. Hypervolemia, excess volume, is a volume increase that leads to increased capillary filtration and reduced reabsorption. It can be due to increased capillary hydrostatic pressure, decreased protein oncotic pressure, and we talked about all these a few slides ago. It increases the risk of edema. We saw that a few slides ago. If there's too much volume, that's going to increase. Remember, we looked at that picture here where there was too much volume here. Too much volume here is going to push that fluid out, and that can cause edema here. So if there's a hypervolemia, that can push the fluid out, increase the risk of edema. It can be hypertonic hypervolemia. In that case, there's an increase in extracellular fluid osmolarity that's going to pull water from the intracellular fluid. So if it's hypertonic, then what you've got going on is that you not only have too much volume, but you have too much volume that has high extracellular osmolarity. And in that case, not only are you pulling it out of the cells, you're pulling it out of the cells and you're putting it into the blood. And so you're going to generate that blood hydrostatic pressure, but your cells are becoming dehydrated because the place you got this fluid was from here, from inside the cells. So the cells are becoming dehydrated because the extracellular fluid osmolality is high. This can be in response to excess blood sodium or blood glucose. That can increase your osmolarity and pull water out of the cells and make the cells dehydrated. It can also be hypotonic. If there's excess free water, then it's not necessarily your solutes. In this case, you've got, if you're hypotonic, you might have excess water. If you have excess water extracellularly, you're going to pull the water inside. But if you still have that excess water outside your hypervolemia, you can still send that water here, which is still going to have the same outcome of pushing it out of the cells. Either way, it's hypervolemia. You've got too much fluid in your tissues. Water intoxication can increase free water. Hypovolemia is too low of fluid, so the volume decreases, the blood pressure decreases. May be compensated. We looked at some of the compensations a few slides ago with the RAS system and the antidiuretic hormone. If your volume goes down, you're going to activate those systems. So the kidneys and the blood vessels can vasoconstrict. You may increase your heart rate if the stroke volume goes down to maintain your cardiac output. So cardiac output will be maintained. So if the volume goes down, the heart rate may go up. So you may be able to compensate for that. But there's a danger if those compensations are prevented by an underlying disease process. So if there's an underlying disease process that's preventing the heart rate from going up, or if it puts too much stress on the heart, or if there's a problem with the kidneys and they can't vasoconstrict, or the blood vessels can't vasoconstrict, this hypovolemia could progress far enough that there can be cardiovascular collapse and shock because there's not enough fluid to maintain that system. It can be hypertonic with dehydration. For example, if there's free water loss. So if you've got hypovolemia and there's an increase in osmolality out here in the extracellular fluid, if that is an increase in osmolality but a decrease in volume, you're really going to pull some water out of those cells, but it may not be able to compensate and the cells are going to become dehydrated. So with dehydration, that can occur because if the cells are dehydrated, then you're only going to be able to keep pulling out far enough depending on how much dehydration you have and where it is. It can be hypotonic. Hypotonic would mean your volume is still low, so you've got a low volume, but it's not hypertonic because you've lost both water and solutes have left. But if the solutes exceed the water leaving, then it may not be hypertonic for that. And that, you could have that if there's an issue with the kidney filtering too much. All right, now we're going to look at some individual actions of ions. So identify the terminology and major consequences associated with abnormal levels of sodium, potassium, calcium, phosphate, and magnesium. When we look at these, we'll get some terminology down first. Hyper means there's an excess, hypo means there's a deficiency. The root nater refers to sodium, and that's why it's a Latin root, that's why it has the symbol Na for sodium. It's a monovalent cation. Cal is potassium, which the kalium has the symbol K for the root kalium, and it also is a monovalent cation. Calcium is a divalent cation, so two positive charges in its ionic state. Magnesium also 2 positive charges in its ionic state, divalent cation. HPO4 2- is hydrogen phosphate. It's a divalent anion, so it provides 2 negative charges. Cations are balanced by proteins in the intracellular fluid and chloride and bicarbonate in the extracellular fluid. All anions, the chloride and bicarbonate, are negatively charged. Cations, of course, are positively charged. Hypernatremia is a serum sodium concentration of over 145 milliequivalents per liter. Its increased sodium concentration will draw water out of cells by osmosis, and that can generate cellular dehydration. It alters the membrane concentration gradient, reduces neuron and muscle cell function. So we have in our cells, we already know that sodium is usually high outside the cell. Well, it's always high outside. I don't want to say always, but the vast, overwhelming majority of the time. Sodium is higher outside of the cells. So if there's an increase in this sodium concentration above what it normally is, that's going to draw water out by osmosis and it's going to dehydrate the cells. But it's also going to alter the concentration gradient. So if we alter the concentration gradient of sodium outside relative to inside, that can change our neuron and muscle cell function. Hypernatremia can be hypovolemic. That means there's low volume. So that'll occur when water loss exceeds sodium loss, so sodium's left behind. So the sodium becomes more concentrated because the water has left and left the sodium behind. So it's hypovolemic. That increases the relative concentration of sodium. Even though the absolute numbers may not go up, the numbers per volume go up. And so the volume has gone down, so the sodium becomes more concentrated. This can occur in uncontrolled diabetes mellitus. What happens is in diabetes mellitus, you may have the glucose out in the blood. There may be a lot of glucose out here in this extracellular fluid. And the water then will follow this glucose. And then if that water then goes to the kidneys and out into the urine with the glucose, then that takes that water out of the body and leaves the sodium behind because it was the glucose that the water was following. There can be euvolemic, which is same volume, loss of free water with near normal sodium. So if there's an intracellular to extracellular volume shift, for example, with excess dilute sweating, that may occur. You may not lose a lot of volume, but what volume you do lose, you might have near normal sodium in that volume. Like if you're sweating, it's dilute, it's still going to have sodium in it, so you're losing some of that, but you're losing the volume, but you're not losing as much sodium. The sodium is still being left behind. Some of it's being left behind, and it still can cause hypernatremia. You can get hypervolemic, increased sodium retention with water retention, but the sodium retention can exceed the water retention, and that might be if you infuse hypertonic saline or if there's an aldosterone disorder, and then the sodium can build up in the fluids, and you're getting the volume too with it. So hypernatremia is an excess of sodium, but it can be accompanied by any volume state, just depending on what's causing it. Hyponatremia is serum sodium below 135 milliequivalents per liter. Can be euvolemic, sodium loss without a net total body water loss. So if there's a reduced sodium intake combined with a diuretic, that can exacerbate the sodium loss. Hypovolemic would be sodium loss exceeding water loss, so reduced relative sodium concentration, because we're looking at hyponatremia now. So you're losing sodium, you're losing water, your volume's going down, but you're losing more sodium, and so your sodium loss is exceeding your water loss, so your water's left behind even though it's a lower volume. So you're reducing your relative sodium concentration because the sodium is leaving more than the water is. It's rare if the kidneys are functioning normally, and something I will say over and over and over is balance is key. If your kidneys are functioning normally, this should balance. The water should follow sodium. But if there's something wrong with the kidneys, then it might get out of balance. This can occur with vomiting, diarrhea with pure water replacement because you're losing more ions than you might be losing more sodium and taking water in, but your volume's still low. Uncontrolled diabetes mellitus. I mentioned that with the last one too, but yes, it can also impact hyponatremia. In this case, the excess glucose in the urine may pull water into the urine as that glucose tubular transport maximum is exceeded, but the water can dilute the kidney, can dilute the sodium. So the balance is the water diluting the sodium and then being lost in the kidneys. That's hypovolemic and hyponatremia. Or is sodium being retained and not lost in the urine with the glucose and water? That was on the last slide. That's hypovolemic and hypernatremia. So it's not just enough to memorize this. If you try to memorize it, you're going to get tied in knots. You've got to understand what's going on and understand what's happening at the cellular level rather than trying to memorize it. Is the water, so let's look at these two. So we have these cells. Is the water diluting the sodium and then being lost in the kidneys? If the water is being pulled out into the cell and diluting the sodium, so we've got lots of water out here and the sodium's being diluted. If the sodium is being diluted, that's hyponatremia. However, if the sodium is being retained, so in this case we have lots of sodium, it's not being diluted, it's being retained, but the water's leaving in the urine. Here the water left in the urine too, following the glucose. Then you have hypernatremia. So I have to give you more information. You can't memorize these. You have to understand what are all my variables. That's another difference between undergraduate and graduate physiology too, is when you took undergraduate physiology, usually you're looking at one variable at a time. In graduate physiology, you're looking at the whole body, because you're going to go out, and when you go out and see a patient, you're not just looking at one system, you're looking at everything, the whole picture. So when we look at the physiology, we're looking at the whole picture. So we can't just say what's glucose doing, what's water doing. We also have to know what sodium's doing. Is sodium being diluted, or is sodium increasing concentration? That's going to depend on what the kidneys are doing too, and what the cells are doing, and what the extracellular fluid's doing, and what you drank, and what you didn't drink, and everything comes in here. Hypervolemic is sodium loss with water retention. So again, uncontrolled diabetes mellitus can cause this. Blood is hypertonic due to increased glucose. Excess glucose in the blood can draw water from the cells into the blood. The blood may become hypervolemic due to water movement intracellular to extracellular, which is going to dilute the plasma sodium, which we saw up here, a dilution. But in this case, the water was lost, and in this case, the water can be retained, not lost, or not lost yet. So we could go hypervolemic and then become hypovolemic. It gets very confusing. So we have to understand what's happening and what are our variables and what are our variables doing relative to one another before we can figure out what kind of condition we're dealing with. I'm much less concerned that you know how to name this and memorize it than you understand what would happen if I tell you something like sodium changes. And I wouldn't say sodium changes. I would say sodium goes up, glucose goes up, water goes up. I would give you the variables and you'd have to tell me what is likely to happen with my osmolarity. So you have to understand this balance and what's going on, and you have to draw out all these conditions and really make them make sense to you. I didn't draw them all out intentionally exactly, because then you will try to memorize my pictures. So I want you to try to draw them out. And that's one of the options that you'll have when we get to class. I'll explain those about the discussion board too. So some of those things I leave open for you to draw for the discussion posts as well. Balance. How much water replacement is there? How much is lost to the kidneys? Is there edema and tissue water retention? That's going to determine if it's hypervolemic versus hypovolemic. You should know what each of those conditions, what would they do? Draw them out. I don't want you to memorize them. I want you to think about them and think about what would happen with those. Understanding is more important than memorizing. Also, one of the things that I'm going to have you do, and it would be in the email that I sent out to you, is write down muddiest points. When you write down a muddiest point, you have to tell me what you know. Because if you just say, could you go through that hyponatremia thing again? I'm going to say, watch the lecture again, because I'm just going to say the same thing I did then. What instead I will say is, you tell me what you know about it, and then we'll see where you're getting stuck. So if you ask me, can you go over it again? I'm always going to say no. Not because I'm crabby, but because I wouldn't say anything different. So how that will go, if you say, can you go over something again? I'll say, you tell me what you know, and then we'll go from there. And so that comes from almost 30 years of teaching physiology. Into the year you graduate, I will be in my 30th year of teaching physiology. It's been a long road. All right. Potassium is the main intracellular fluid cation. The diffusion of potassium out of excitable cells makes the cell interior negative relative to the extracellular fluid. And I don't actually have that drawn on here, but sodium goes out of the cells, makes the interior negative. That's what causes repolarization. The cell concentration is then regulated by the sodium-potassium ATPase, and that's going to bring that potassium back in. So potassium coming back in, sodium going out, is the major determinant of the resting membrane potential. This regulates the cellular electrical neutrality in relation to changes in hydrogen and sodium levels too. In the intestines, when potassium is absorbed, it's going to stimulate insulin release. That's going to activate this up here. So when insulin is released, that's going to activate potassium absorption into muscle and liver cells and co-transport with glucose. What that does is it prevents an acute hyperkalemia related to food intake. Our body has these ways of maintaining balance, and we'll say, well, that's really weird. Why is potassium going in with glucose? What possible reason could it have inside the cell for wanting to get in there? And that's the wrong question. The reason is so that it doesn't build up in the blood as it comes in from the nutrients. Otherwise, it could quickly cause a hyperkalemia. So it goes into the cells in order to balance the blood. Insulin deficiency then would shift the potassium out of the cells and alter the membrane potential, because it's going to lead to an hyperkalemia. It can stimulate aldosterone release, and that's going to increase potassium loss in the urine, which doesn't seem like it makes a whole lot of sense. We're going to absorb the potassium in the intestines just to get rid of it in the urine, but it's all about maintaining that balance, because we need to absorb it in the intestines because we don't know how much we're going to need, and then any excess we can get rid of in the kidney. So we take it all in and get rid of what we don't need. If there's an aldosterone deficiency, then we're not going to get rid of the potassium, because we would not be able to counter-transport it with sodium, and again, we're going to alter the membrane potential. In acidosis, the potassium's going to move out. Remember, potassium's high intracellularly, but potassium is a monovalent cation, and so is hydrogen. So one of the things we can do to be protective of our cells, our extracellular fluid in acidosis, is we can push potassium out in exchange for hydrogen coming into the cell, and that gets the hydrogen out of the extracellular fluid and helps to control that acidity in the extracellular fluid. The problem is then there's a danger of hyperkalemia in the blood. So it can increase the risk of hyperkalemia and alter the membrane potential. So hypokalemia, if serum potassium falls below 3.5 milliequivalents per liter, that can result in decreased extracellular fluid potassium. So potassium moves from the intracellular fluid to the extracellular fluid. So with hypokalemia, normally most of our potassium is intracellularly, but we do have some potassium extracellularly. If this goes down, then we're going to pull some potassium out to balance that decrease, because we're going to it's going to change the flux gradients and pull some out through the leak channels. The loss of those intracellular cations, these are carrying a positive charge, is going to make this even more negative inside the cell. It's going to cause the cell membrane to hyperpolarize. If the cell membrane hyperpolarizes, then we need a stronger stimulus to reach threshold for membrane depolarization. So this is our action potential graph here. This is normal. This is our membrane potential. This is our threshold. We saw this graph before, that we need to reach from our membrane potential to threshold to generate this action potential that's all or nothing. Now, if we have low potassium outside the cell, that's going to pull potassium out, and we're going to hyperpolarize, which means our membrane potential is going to get even lower. That means we have a bigger gap to bridge before we reach threshold, before we can generate an action potential. So we're hyperpolarizing, making it more polar, making our cell membrane more polar. To get that cell to depolarization is going to take more of a stimulus. So it's more difficult to generate an action potential with hypokalemia. The outcome is the skeletal muscles are going to get weaker. We're going to have atony. We can get to cardiac dysrhythmia. Remember that when we administer insulin, that we're going to stimulate the potassium movement from the blood into the liver and muscle cells. That can generate some hypokalemia, depending on how much insulin there is. We may have reduced potassium intake with alcoholism or nutritional deficiency. Alkalosis. In alkalosis, that means, that's terrible, that means the hydrogen is too low. In that case, we're going to pull some hydrogen out of the cell, and we're going to push some potassium in, and then that will decrease. That can cause hypokalemia. So alkalosis can cause hypokalemia. That's going to risk changing the membrane potential. Diuretic treatment, if it increases the potassium clearance, that can decrease potassium in the blood or in the extracellular fluid. Any volume depletion, remember that RAS system. If you deplete, if your blood pressure falls, if you lose any fluid, you're going to stimulate RAS. And one of the things that happens when you stimulate RAS is that you release aldosterone. Well, aldosterone stimulates sodium retention but potassium secretion, and so you decrease your extracellular fluid potassium as an outcome of that. Any of those things can generate hypokalemia and make it more difficult to generate an action potential. We don't get too much into ECGs, but I do want to mention them here because your book does. You'll have a whole ECG class starting in October. And so we'll look at ECGs in the cardiovascular system a little bit, but I do want to mention them here because your book does mention hypokalemia and hyperkalemia, and I do get some questions about it. So in a normal ECG, the P wave is atrial depolarization. That is caused by a sodium influx into the atrial myocardial cell, so a depolarization. QRS complex is the ventricular depolarization. That's going to mask atrial repolarization. Sodium influx into the ventricular myocardium cells. Potassium efflux out of the atrial myocardium cells. The T wave is ventricular repolarization. Potassium efflux out of ventricular myocardium cells. So if we look at this normal ECG, normal P wave is sodium influx. QRS, sodium influx. Ventricles, this was atrium. And potassium efflux, atria. And T wave is potassium efflux, ventricles. The thing I want to mention is we've already looked at, and I'll mention a little bit more about ECGs here, but something else that's important to remember. We looked at the action potential. This is one cell. This is millions of cells combined. And it includes not just cells, but direction. ECG depends on direction. It depends on where you place the leads. What direction is the charge flowing? So don't confuse these for one another. They're very different things. They are, oh, shoot. I messed up my pen. Oh, good. Whew. Sometimes it takes me forever to figure out what I did wrong when I push buttons. All right, so it's not the same thing. Not the same thing. If the cardiac membrane hyperpolarizes, now we talked about low potassium in the neuron here with the action potential, but this could also happen if this is a cardiac cell. If there's low potassium in this cardiac cell, you're going to need a larger signal here for the action potential to occur too. So in hypokalemia, that can occur where you need a larger than normal action potential. That can reduce your action potential frequency. A decreased extracellular potassium would be expected to pull potassium with a greater gradient to extracellular fluid for faster repolarization. Think about what that means. So if I have lower potassium out here, and I have potassium in here, you're thinking, okay, well this is really low, so that means this is a bigger hill. This is really high, and that's not really high, but it's high, you know, it's normal. But that makes it a bigger hill, so I'm going to pull that really fast out of here and repolarize that like crazy. That's what you would think would happen, but remember that picture I showed you a while back, that the myocytes have 7 main types of potassium channels active under different charges and concentration gradients, so it's really difficult to pinpoint the exact impact of hypokalemia on the cardiac muscle. There's a paradoxical channel called a delayed rectifier channel, which actually goes the opposite direction. That was the one I circled that showed the opposite direction. That actually reduces potassium transport under conditions of decreased extracellular potassium, and it may end up prolonging the cardiac action potentials, which is completely not what you would expect based on this. So when I say that, it's a much more complex picture than what you would have learned in undergraduate physiology. Now, some of you may have a lot more cardiovascular physiology if you took extra cardiovascular physiology, or if you have more experience in cardiac care. But for regular general undergraduate physiology, the picture is a lot more complex. There's a lot more variables than what's usually learned. Potassium can also be tied to arrhythmia through interactions with calcium. If there's a decreased extracellular potassium, so let's draw our cell up here again. So we've got lower potassium here. Remember that the sodium-potassium exchange transporter, the sodium-potassium ATPase, usually is pulling out pulling out 3 sodiums and putting in 2 potassiums to reset that gradient. But if there's not enough potassium here, then there isn't enough potassium to go into this transporter, so that's not working either. So there's a decreased sodium efflux in this transporter because there's not enough sodium to fuel the pump, or potassium, I'm sorry, to fuel the pump. But there's another channel, the NCX channel, that can also have sodium pulled out with calcium moving in. So there's another channel that can have an impact there too. That's going to impact the ECG. If there's an increase in this intracellular calcium and decreased intracellular sodium, because it's left here, then those can be associated with hyperpolarization, and that's where you get this big U wave with hypokalemia on the ECG. Calcium's a positive ion, but it's normally sequestered in sarcoplasmic reticulum, so if it builds up, it can have an impact. The actual effect is often a longer action potential because of the paradoxical delayed repolarization channels, the potassium repolarization, and altered sodium and calcium, so there's an increased risk of arrhythmia. What I want you to get from this is not these big ECG changes, but if I don't mention it, then people always ask about it, because a lot of people have experience with a cardiac and they focus in on this hypokalemia. So I want to explain what we know about it to kind of try to forestall some of those questions on those details. Not that it's not important, but you're going to get a lot more on this when you take your ECG class. So this is just a very basic, why is this happening physiology. You'll get a lot more in ECG. Thinking of it in terms of a simplified potassium channels repolarized mechanism is not necessarily going to align with reality. The picture's a lot more complex. There are 7 potassium channels that do 7 different things under 7 different conditions. And so it can get really tricky when it comes to potassium and the heart. Hyperkalemia is a serum potassium over 5.5 milliequivalents per liter. It increases extracellular potassium. It's rare because of the renal secretion of potassium can occur at several locations in the healthy nephron. So we have a lot of ways to get rid of potassium in the nephron. So unless the kidney's messing up, it's probably not going to happen. Cell trauma, though, can increase potassium because remember that potassium is high intracellularly. So if there's damage to the cell, that potassium can leak out. If the cell membrane gets damaged or if the cell gets crushed or anything like that, that can leak out. We also may get that intracellular to extracellular shift with acidosis or hypoxia or insulin deficiency, all those things we've looked at before. We didn't look at hypoxia, but hypoxia, remember that we have this sodium-potassium channel, and the sodium-potassium channel is pulling 3 sodiums out to reset these gradients and pulling 2 potassiums in. But if you have hypoxia, you can't generate, if you have low oxygen, you can't generate as much ATP, and you need ATP to work this channel. So with hypoxia, you can't get that channel functioning, and so that's going to increase activation of, we have another type of potassium channel that's dependent on ATP, and that's going to further increase those potassium levels because you're not pulling them in here, but you're going to get this impact of this. So it's also another thing that has to be balanced, to keep in mind that hypoxia can do that. Reduced production of aldosterone may reduce potassium secretion. What happens with hyperkalemia, high potassium, is because the potassium is high here, and it's going to be high then in the intracellular fluid too, then we're not pulling it back in this way, but we alter our gradient. So what happens is that we actually, with these positive ions, pull this membrane closer to 0. So we don't have as much of a gap before we get to threshold. We pull this membrane up our normal threshold, our normal membrane excitability closer to threshold, and we only have this little gap. So any stimulus that bridges that little gap is going to send us into an all or nothing action potential. So it brings it closer to threshold. So there's less gradient drawing the potassium out of the cell, less stimulation needed to reach action potential threshold. That increases the excitability. It decreases the ability to fully repolarize, though, because once that sodium comes, or the potassium comes back in for repolarization, so here we have depolarization, repolarization, it only goes this far instead of going all the way down here, so it doesn't fully repolarize. It stays right at that level that's very close to that threshold. So not much of a gap there. The actual impact depends on the severity of the imbalance for hyperkalemia. If it's mild hyperkalemia, so here's our high hyperkalemia here, the cell will reach threshold more quickly and repolarize more quickly, but it's not going to repolarize it. Well, it's higher. It's going to go all the way up here, but it's not going to repolarize all the way. It increases neuron and muscle cell excitability because there's just that little gap between the membrane and the threshold. If it's severe, then if the resting membrane potential actually exceeds the threshold, so if it's so severe that this membrane potential is actually above it, then you can't repolarize at all because you can never get back down that far. And then you're going to have muscle paralysis and cardiac dysrhythmia because the membrane potential is above the threshold, so it's always going to be depolarized. It can never repolarize in that case if it gets up that high. The impact may be altered by relative calcium concentrations. So calcium also has a role. Calcium alters the threshold. So with calcium, here we've got calcium. The membrane potential is in the normal spot, but the threshold changes based on the calcium. So if there's high potassium, so here's high potassium and low calcium, there's increased excitability because now look at your membrane. If we pull these two together, your membrane potential is like right at your threshold if you've got these two conditions going on. And so your cell's always going to be in excited condition. If you've got high potassium and high calcium though, if you've got high calcium, that raises your threshold and that would normally decrease your excitability. But if you've got high potassium, that actually gives you a normal gap there. So that's a little protective if those two are both high. So it depends on what each one's doing relative to another as well, just to make the picture a little more complicated. You know, we'll throw some other ions in there. That's what I'm saying though. You've got to look at, if you're used to looking at one system at a time, now we're going to mishmash all those systems together and we need to figure it out because the body is not separate boxes. The body is all one integrated thing. One integrated system, and everything impacts everything else, and it's time to bring it all together now. When we look at calcium, 99% of the body calcium is in bones as hydroxyapatite. The remainder is in plasma and cells. Calcium is a fundamental metabolic ion. It's an enzymatic cofactor, an intracellular signaler. We saw it already with the inositol triphosphate system. It's important in muscle contraction. We'll look at that in future units. A major player in the body, calcium. Its concentration is regulated by a balance release of parathyroid hormone from the parathyroid glands, which increases blood calcium, calcitonin from the thyroid glands, which decreases blood calcium. The calcium that's circulating in plasma is about 55% bound to plasma proteins, or albumin or other compounds, about 45% free or ionized, and that's the most important physiologically because that's what's going to have an impact with these enzymes and these muscles and all that. The fraction free is impacted by pH. Everything impacts everything else. Acidosis increases ionization. Remember from previous slides that acidosis, potassium moves from the intracellular fluid to the extracellular fluid. So in acidosis, in order to counteract this increase in hydrogen, we move that into the cells, and potassium moves out of the cells. They both carry positive charge. That's going to balance the hydrogen movement. Also recall from the last slide that a high potassium combined with a high calcium is protected. So that gives us almost a normal gap here. So it's protective for our neurons. What that tells us is that these physiological systems are tightly linked to maintain homeostasis. Balance is key. Balance is key. This integrates a lot of systems and it gets really complicated, but fortunately, this one's a little bit easy to keep straight. Acidosis is an increase in extracellular hydrogen. As acidosis increases, it's associated with an increase in extracellular potassium. We saw that here. As hydrogen goes up, we put some hydrogen into the cells and potassium goes up in the extracellular fluid. It also is associated with an increase in calcium ionization. So increased ionization in the blood. So at least they all three go up together. So that's pretty easy to remember. Alkalosis would have the opposite effect. It increases protein binding of calcium rather than ion release. Next up, we'll look at phosphate. 85% of the body phosphate is in bone as hydroxyapatite. So that's in combination with calcium. It's also in phospholipids, nucleotides, DNA, and ATP. The P is phosphate, and in ionic form. Calcium and phosphate regulation are inversely proportional. When calcium's low, you don't want it combining with phosphate to make bone. You don't want hydroxyapatite if your calcium's low. So if the calcium's low, calcium's reabsorbed and phosphate is excreted. The parathyroid glands increase parathyroid hormone release when calcium's low. So the parathyroid glands up here, if calcium is low, we increase parathyroid hormone, and that will work to increase calcium then. Parathyroid hormone works at the kidneys and intestines to increase blood calcium. Stimulates osteoclast formation, so it will stimulate a hormone or signaler called rankle, which stimulates, the most important thing about this is it stimulates the osteoclasts, which increase calcium release. So it's going to release calcium from the bone into the blood. Vitamin D is a fat-soluble steroid ingested or formed in skin upon exposure to sunlight. If calcium decreases, so if you have a decrease in calcium, that's going to increase parathyroid hormone. That stimulates renal calcium absorption. So parathyroid hormone is going to go to the kidneys, and it's going to cause an increase in calcium reabsorption into the blood. So it's going to stimulate calcium reabsorption. It's going to cause an increase in calcium in the urine. It's going to inhibit phosphate reabsorption. Oh, I messed that up. I'm thinking ahead of myself. We don't want to increase calcium in the urine because we're increasing calcium in the blood. It's going to increase phosphate in the urine. There we go. So it's going to inhibit its reabsorption. The combination of this low calcium and high parathyroid hormone stimulates renal vitamin D. So it's also going to stimulate vitamin D3 activation from D3. Vitamin D3 stimulates the absorption of calcium and phosphate in the small intestine. So here's our small intestine. And it's going to stimulate calcium and phosphate into the blood. Increases bone calcification, so it's going to pull that calcium into bone. I don't have that on this one. Increases phosphate excretion in the kidneys. So it absorbed it here. Now it's going to put it over here. Now that may seem totally counterproductive. It absorbed it in the intestines, but now it's going to get rid of it. Vitamin D3 is going to stimulate getting rid of it in the kidney. But remember that it doesn't know how much it's going to need, so it absorbs it all from the diet and then gets rid of what it doesn't need. Balance is the key. Renal failure then is going to result in decreased blood calcium because it's going to inhibit all of these things. So it's going to result in decreased blood calcium and increased phosphate because there's not going to be as much D3. And it's not going to respond to that parathyroid hormone as well either. So a lot of interactions going on with calcium through parathyroid hormone and also through vitamin D. Calcitonin is formed by C cells of the thyroid gland. Calcitonin levels increase when calcium's high. So here's calcitonin down here. Increased calcium increases calcitonin. It inhibits parathyroid hormone release because calcitonin wants to keep the calcium in the bones. It reduces renal vitamin D activation, so it's going to oppose everything that's on the previous slide. We're going to reduce intestinal calcium absorption, increase renal phosphate absorption. It's going to just oppose everything on the previous slide. And then the thing it does on this slide is calcium is going to inhibit this breakdown of osteoclasts. So it's going to prevent that loss of calcium from those osteoclasts. So it's going to retain the calcium in the bone, the calcitonin. And that way it can decrease the calcium in the bone. Hypocalcemia may result from a wide variety of issues. Thyroidectomy, when you take out the thyroid gland, you may also lose the parathyroid gland, so that's going to reduce parathyroid hormone, and that will reduce the intestinal calcium reabsorption from the slide a couple slides ago. The diet, if there's a reduced calcium intake, increased phosphate intake. Remember that phosphate can bind to calcium in the intestines, and then it will prevent the uptake of both. Reduced to sun exposure, if there's a reduced vitamin D3 activation, then that's going to decrease the amount of calcium that's retained from the picture a couple slides ago. If there's a bone neoplasm, that causes an increased calcium deposition in bones or in the soft tissues, that's going to decrease the serum calcium because it's pulling that calcium out of the blood to put it into the bone or into the soft tissues. Alkalosis. Remember that alkalosis increases plasma protein binding of ionized calcium, so that's going to reduce free calcium. For alkalosis, we are going to get the other side of the relationship: low hydrogen, low potassium extracellular, low calcium ionization. That's our alkalosis relationship. Blood transfusion, that's going to depend on the concentration of the calcium chelators, which are the preservatives in the blood transfused that can bind to the serum calcium. It also may reduce the availability of calcium in the transfused blood because it doesn't want that transfused blood to clot. So they'll chelate that calcium so that it's not generating, because calcium is a big activator of the clotting cascade, and so that can impact serum calcium too. So with hypocalcemia, we have a reduced extracellular fluid calcium. That's going to pull calcium out from the intracellular fluid through that sodium-calcium exchange pump. Remember, I mentioned that we had this sodium-calcium exchange pump. The thing about the sodium-calcium exchange pump is that it can go in either direction. So if our calcium is lower outside, then we can pull more calcium out from the intracellular fluid through this NCX pump. But that's going to force sodium in because it has to be antiport. It can go either direction, but it has to be antiport. So if we pull calcium out, we have to pull sodium in. So if calcium's low in the extracellular fluid, we can pull some out of the intracellular fluid. But that's going to increase the intracellular fluid sodium, and when we increase the intracellular fluid sodium, that's going to partially depolarize our cell. And then if we partially depolarize our cell in low calcium, then that makes our cell more excitable because it decreases this gap. It's easier to get to threshold then, so by pulling that sodium in. That can lead to muscle spasm and tetany. Now, normally also, in our voltage regulated sodium channels that sodium normally comes in, calcium actually blocks those. But we have reduced calcium, which means we have a reduced calcium block, which means more sodium can come through. And that's even going to make it more depolarized. So we even have this gap lower, and that contributes to making that excitability even more. So reduced extracellular calcium reduces that sodium block, and it's easier for sodium to come in and cause depolarization. That also can lead to muscle spasm and tetany. It can delay the cardiac action potential, increase the risk of prolonged QT interval and arrhythmia, because it's altering that excitability not just of the nerve cells, but also of the cardiac myocytes. Hypercalcemia may result from hyperparathyroidism as a consequence of thyrotoxicosis. So if the thyroid gland is overactive, that can release more hyperparathyroid hormone, or more parathyroid hormone, because it's hyperparathyroidism, and that's going to increase calcium reabsorption. Excess vitamin D can increase calcium reabsorption. We can have increased calcium reabsorption from bone, cancer metastasis from another site like breast. In this case, if there was the calcium metastasis from breast, that might increase the calcium reabsorption from the bone depending on the mechanism of those cells, versus the calcium neoplasm we saw before that could increase the deposition into bone. Prolonged immobilization is a common one because then we're constantly remodeling our bone in response to weight-bearing stress, and if we remove the weight-bearing stress, then the bones are not going to be as strong, and so we lose that calcium, and that could go into the blood. Acidosis reduces serum binding to calcium. Remember that the relationship when hydrogen goes up in the extracellular fluid, so does potassium, and so does calcium ionization. So it reduces the binding to albumin. So we get increased ionized calcium levels. Hypercalcemia will elevate the extracellular fluid calcium, increase extracellular fluid. Remember I mentioned that we had that sodium channel here. That sodium's coming in, that's blocked by calcium. Well, in this case we have high calcium, so we have more of a block. So what happens with high calcium is the opposite of what happened with low. It makes that threshold gap bigger, so it's harder to overcome that. It makes it less excitable. So the membrane potential and the threshold become further apart, and that increases the threshold, that gap. So it's more difficult for the cell to reach threshold for depolarization for an action potential. The skeletal muscle weakens. There's a shortened cardiac QT interval once that action potential does occur, and that can lead to arrhythmia. The increased extracellular calcium, so with this calcium high out here, remember I said that this sodium calcium pump can go either direction. If the calcium is high, that can pull the calcium in and then pull sodium out, because that can flip. As long as they're going in opposite directions, it can flip. That's going to draw sodium to the extracellular fluid in exchange for calcium to the intracellular fluid, and that can even make that gap bigger because it's going to hyperpolarize the cell. Then you're pulling a positive ion out. That's going to hyperpolarize it, and even though calcium is positive, then that calcium would normally be sequestered into the sarcoplasmic reticulum if this is a muscle cell. So, but then there's more calcium available to increase that force of contraction when you finally do reach depolarization, so you can get a stronger contraction once you finally get there. Let's look at phosphate. Hypophosphatemia is a reduced extracellular fluid phosphate concentration. That can be due to reduced intestinal absorption, due to malabsorption disorders. Antacids, magnesium and aluminum, can bind to phosphate and decrease their absorption. Alcohol abuse can reduce nutrient intake and reduce phosphate. It can be due to increased renal excretion. If there's hyperparathyroidism, the parathyroid hormone inhibits renal reabsorption, so you can lose too much phosphate. It can be metabolic alterations. Increased cellular uptake of phosphorus is used to produce phosphofructokinase. When we start the process of glycolysis, we add phosphate on, and so we need phosphate for that. So if metabolism goes up, we need to draw more into the cells to support that if metabolism increases. After starvation, phosphate's going to enter the muscle cells to replenish the creatine phosphokinase, which is a storage form of energy, and so the blood phosphate levels can fall because of that. Hypophosphatemia results in the reduced production of red blood cell 2,3-diphosphoglycerate, DPG, also known as bisphosphoglycerate, BPG, so it can be written as both of those ways. The 2,3-DPG binds preferentially to deoxyhemoglobin, so if there's hemoglobin that doesn't have oxygen bound, the DPG or BPG will bind to the hemoglobin, and what that does is it changes the shape so that oxygen is released from the hemoglobin, And so it prevents the oxygen from binding at low oxygen levels, which is good because low oxygen levels are usually out in the tissues. And so it increases oxygen release out to the tissues for metabolism. So we want this. This is a good thing to decrease the oxygen binding because it releases that oxygen release out to the tissues. But if there's low 2,3-DPG, it's going to prevent that oxygen release, and the oxygen is going to stay bound to the hemoglobin then, and that's bad because oxygen bound to hemoglobin is not doing anything for us. It's just circulating around on the merry-go-round, not ever getting off the horse on the carousel. And so it isn't released to tissues because the oxygen's still bound to hemoglobin. So we need that 2,3-BPG or 2,3-DPG to get that oxygen released. When that's released, and this will be a excuse me, picture that's very familiar to you, as DPG is decreased, then our saturation of oxygen goes down at any given pressure. And we don't want that. We don't want our oxygen levels to go down, our saturation, because that's a measure of how much oxygen is available to be released to the tissues. And we want the tissues to have that oxygen. So we need to get that out there. If we don't have the oxygen out there, we can't produce ATP because we can't go through the full process of electron transport chain to clear out that hydrogen. Increases the risk of hypoxia, muscle weakness, reduced central nervous system function because we can't generate enough oxygen release to get that ATP production going. Bone weakness may also occur if phosphate's lower because there would be increased phosphate reabsorption out of the bone to the extracellular fluid to try to make up that balance, and then the bone is going to be weaker. Hyperphosphatemia is elevated extracellular phosphate. Can be due to cell destructions. Phosphate is more of an intracellular thing. If it moves from intracellular to extracellular, for example, with any cell destruction, such as cancer chemotherapy, then that can move that phosphate out and increase it in the extracellular fluid. If there's a reduced glomerular filtration rate, phosphate's filtered at the glomerular capillaries, and so it can be retained in blood if it's not filtered. If there's hypoparathyroidism, so a lower parathyroid gland, then if the parathyroid hormone is not there to inhibit the reabsorption, it can be increased in the renal tubule, the reabsorption, and increase in the blood. This can result in increased hydroxyapatite formation because it can combine with the calcium and then be deposited not only into bone but also possibly soft tissues as well if there's too much hydroxyapatite. That's going to reduce the serum calcium because it's going to sop up. To make hydroxyapatite, you need both phosphate and calcium. So that's going to bind those together, and then you're not going to have the calcium either. So you're going to have all this bone formation, but it's going to decrease your calcium. Remember that calcium blocks those sodium channels. So if we don't have that calcium to block those sodium channels, then sodium is going to increase the frequency of action potentials. So in that way, if there's too much phosphate using up the sodium or the calcium can't block the sodium, so it's also going to have muscle impact. Very indirectly, but it's still going to have muscle impact. Magnesium, 40 to 60% of magnesium is in the bone with the rest in cells and plasma. Plasma is about 1/3 bound to plasma proteins with the rest in ionized form. If magnesium levels fall, renal magnesium conservation will occur. It will try to conserve it. In cells, it's the second most abundant intracellular fluid cation after potassium, so magnesium in the cells. The functions of magnesium. Magnesium is kind of a tricky one. It's very reactive to calcium. It's a cofactor in intracellular enzymatic reactions. Things like you need magnesium for nucleic acid stability, protein synthesis, neuromuscular excitability, metabolism. It is a crucial element. It's used in so many things. But it often interacts with calcium. It's frequently antagonistic to calcium, but its effects may appear to mimic one another. There's a delicate balance, and calcium and magnesium are something I'm still researching, even all these years, because it's really tricky to tease those out from one another. They're not, the research hasn't been done to give a really clear picture of why these are interacting like that and how exactly they're interacting. Hypermagnesemia is elevated extracellular fluid magnesium. It's rare. It's usually secondary to renal failure. Magnesium inhibits the inward rectifier potassium channels. We talked a little bit about those. Those were ones that fixed that potassium to give it that impact on that cardiac, you know how we talked about the cardiac effect if the potassium is altered, and so magnesium normally inhibits that. So if magnesium's high, then the efflux of potassium out of the cell exceeds the potassium influx, and the cell can hyperpolarize and reduce its excitability. This can stimulate the sodium-potassium ATPase function that supports the resting membrane potential. All of these ions are interacting, so there's this balance. So if there's this hyperpolarization, but then magnesium stimulates sodium-potassium to try to fix that, then there's this balance. So that's why I say it's really hard to figure out magnesium, because there's so many things we don't know that it's involved in yet, or they're still trying to figure out. So it's hard to give a good magnesium picture. It inhibits L-type calcium channels, so it may impair the plateau of the cardiac action potential and reduce contractility in the heart. The calcium versus magnesium, it's a delicate balance. The absolute ion numbers are not as important as the numbers are relative to one another. So it's not just enough to say magnesium is this level. You have to know magnesium is this level relative to calcium, which is this, is calcium up or down, and it gets pretty involved. The increased magnesium will mimic hypercalcemia, though. Reduced magnesium can mimic hypocalcemia, so that even makes it trickier. Are they opposing each other? Are they mimicking each other? It's tricky to try to figure out magnesium, and I'm still working on the magnesium part. Hypomagnesemia is reduced extracellular magnesium. This one's a little more clear. We know a little bit more about what happens if magnesium is reduced. It can happen due to nutritional deficiency, if there's a kidney tubule dysfunction with increased magnesium loss, altered metabolism like with diabetes. We know that magnesium inhibits the L-type calcium channels, and then low magnesium is a little clearer. There's a loss of that channel inhibition, so there's an increase in calcium flow through those channels, and that results in increased muscle neuron irritability and tetany. And so one of the main outcomes of low magnesium is muscle cramps because of this. It can also be associated with hypertension because of the vascular tone. The blood vessels aren't relaxing. So muscle cramps and hypertension are common with hypomagnesemia because of that impact through the calcium channels, but still trying to figure out how exactly those two are interacting. Objective 5, review acid-base homeostasis and associated buffering systems. Hydrogen ions are a byproduct of metabolism, and they must be neutralized. We saw that before. Glucose is broken down in metabolism into carbon dioxide and hydrogen and ATP, well, energy, not directly ATP. We have to make the ATP. So energy. We need to get rid of this hydrogen. Carbon dioxide we're going to breathe off, but we need to get rid of this hydrogen as well. It needs to be neutralized. Volatile acids are respiratory gases, and with hydrogen, we generate this carbon dioxide in the tissues, and it's not just this hydrogen that goes to the NAD we have to worry about, but also this carbon dioxide that we're going to breathe off. Before we breathe it off, it actually will combine with water to form carbonic acid. And so a lot of times that's really confusing. When people are studying physiology, they'll refer to carbon dioxide as an acid. Well, if you've had chemistry, you're looking at it going, there's no hydrogen on carbon dioxide. How can that be an acid? Well, the reason it's an acid is because it combines with water using the enzyme carbonic anhydrase, and it combines to form carbonic acid, which then dissociates into hydrogen ions and bicarbonate. So it's not just the acid that we're making here, we also make it here from the output of carbon dioxide. So all of that has to be dealt with. And then at the lungs, though, we can take this back this direction and then breathe off. That carbon dioxide is breathed off at the lungs. So we can get rid of it that way, and then it's just converted into carbon dioxide and water. We also make non-volatile acids. So during the process of metabolism, we can make sulfuric acid, phosphoric acid, lactic acid, pyruvic acid, keto acids. All of those can also be produced. And those are either eliminated by the kidney or metabolized in the liver. Hydrogen concentration is measured by the pH scale. 0 is the most acidic, 7 is neutral, 14 is alkaline. Arterial blood is slightly alkaline at pH of 7.4. Below 7.35 is acidosis, and you can see that on this picture. And above 7.45 is alkalosis here on that picture. Venous blood is normally around 7.35. Venous blood's a little bit more acidic because it has more carbon dioxide, because it's taking that carbon dioxide away from the tissues back towards the lungs. Arterial blood is headed out towards the tissue, so it doesn't have as much carbon dioxide. So it doesn't usually have such a low pH. The pH of other body fluids varies based on buffer status and metabolic function. So stomach pH is low, pancreatic is higher because it has a lot of buffers. A pH change of 1 indicates a hydrogen concentration change of 10. The scale is logarithmic. So pH is log of the fraction, or a negative log 10 of hydrogen. Because it's the log of a fraction, it's an inverted scale. That means that as acidity increases, pH decreases because we're measuring the log of the fraction. So as this number gets bigger, this fraction gets smaller, closer to 0. So the closer to 0 we get, the lower the pH is. Hydrogen homeostasis is crucial to maintenance of normal enzyme function. So we have buffering systems that occur in response to acid-base changes. Buffers will absorb excess hydrogen or release it as needed to reduce the pH fluctuations from normal. Our main buffer systems in plasma are hemoglobin and carbonic acid. Hemoglobin, you can see that one here. Hemoglobin will pick up a hydrogen, and if it also is picking up a carbon dioxide, depending on how much oxygen is in there, in this case it's got a carbon dioxide, and so you get the hemoglobin with the acid and the carbon dioxide attached. But it could also just be the H hydrogen on the hemoglobin if it's got oxygen attached. But that will pick up a hydrogen on a hemoglobin and help to buffer it. Carbonic acid is, I'm losing my voice, we're at 3 hours and 23 minutes. Last year this was 4 hours and 6 minutes, so I'm trying to get it under that here. All right, so I did cut out about 9 slides, but they'll come back later in later units. All right, so the carbonic acid system is when we take in that's hard to see, I'll write it up here. Carbonic acid is water plus carbon dioxide converted into carbonic acid, which is broken into hydrogen and bicarbonate. And there's an enzyme that takes care of that, carbonic anhydrase. And so that acts as a buffer system, because if hydrogen goes up, we can take it in this direction and kind of buffer it and convert it into carbon dioxide and water. But that's only going to be effective if we can get rid of the carbon dioxide through breathing it out. In intracellular fluid, if we're inside the cells, we can use proteins and phosphate. And this one actually has that written in the plasma for proteins, which it can also occur in the plasma, where the proteins can absorb that extra hydrogen onto their amine group here and release a carbon dioxide and take that hydrogen on there. Now that's only going to be effective though if that carbon dioxide doesn't then immediately go into the carbonic acid cycle, or carbonic acid cycle, carbonic anhydrase enzyme. Proteins have a negative charge, so they can bind and release the hydrogen as needed. Also, there's a phosphate system, so phosphate can pick up the hydrogen and convert into the dihydrogen form. In the renal system, also that phosphate, and also an ammonia system. The ammonia can pick up an extra hydrogen and be converted into an ammonia ion. The pK value is the pH at which a buffer pair is half dissociated. So this is, all of these buffer pairs have pHs where they're most effective, and if the pH is not balanced at that pK, then it might go one direction or the other, and so it may not be an effective buffer at that pH. An example is the pK of the carbonic acid bicarbonate. So right here, this conversion here, bicarbonate and carbonic acid, its pK is 6.1, so it's balanced at 6.1, but the blood is 7.4, so it seems like that wouldn't be an effective buffer system, but it is, as long as, like I said, we keep getting rid of that carbon dioxide, and then that pulls that in this direction, because we're getting rid of this product of carbon dioxide, and it can keep the buffer moving in this direction. And so we can keep clearing that hydrogen out. Compensations occur when adjustments happen or occur in response to primary changes in pH. This is different than a correction. If there was a correction, then the values for both components of the buffer pair would return to normal. But in a compensation, we're just changing something else to compensate for that change in pH. Compensation can be cellular ion exchange. We've seen this before. So if hydrogen goes up in the extracellular fluid, so it's acidic, we can push the hydrogen into the cell and pull some potassium out, but that's a risk of hyperkalemia then. So in acidosis, hydrogen enters, potassium leaves. That may lead to hyperkalemia, and then the opposite would happen in alkalosis. The hydrogen would leave and potassium would enter, and that can lead to hypokalemia. There can be respiratory compensation, which involves changes in ventilation rate. So there are central chemoreceptors that monitor pH and the arterial concentration of carbon dioxide, arterial pressure of carbon dioxide. In acidemia, the compensation is to increase the carbon dioxide that's given off, because we're going to go in this direction here and we want to get rid of that carbon dioxide, and so that's our compensation. In alkalemia, we want to decrease the ventilation rate, because then we can go in this direction. We can retain the carbon dioxide and go in this direction and then build up some hydrogen to counteract that alkalosis. If we look at renal compensation, we get changes in urine pH. In the distal convoluted tubule, we can secrete into the renal tubule cells some carbon dioxide and water. And when we do that using carbonic anhydrase, we can make carbonic acid and we can generate hydrogen and bicarbonate. If we regenerate that bicarbonate and take that back to increase the blood pH, we can compensate for the acidemia, the increased acid in the blood, and then we regenerate that bicarbonate, and then we can increase the urine acidity. But we don't just pump hydrogen into the urine. What we do is we have this phosphate that's filtered at the renal tubule, and that can pick up hydrogen and form the dihydrogen form, and then that can be lost in the urine. And we also, so that can combine with hydrogen released to the urine, and then we also have ammonia, and that can be, and I'm not sure of my ammonia and ammonium. I didn't look those up before I started, so I'm not sure which one is which. But that is lipid soluble and can be taken through the cell and then into the filtrate and then that can also pick up a hydrogen and be lost in the urine. So in that way, we can get rid of those extra hydrogens as they bind with the phosphate and the ammonia, and then increase the urine acidity. So that can help compensate for acidosis as well. Objective 6, compare and contrast the causes and compensations of metabolic and respiratory acidosis and metabolic and respiratory alkalosis. Acidosis and alkalosis may each be either respiratory or metabolic. Is the pH low? So are we going in this direction? That's acidosis. Is the pH high? Alkalosis. So if the pH is low, the next thing we look at is what's the carbon dioxide doing? Is the pressure of arterial carbon dioxide high? That means we're retaining too much, so it's respiratory, usually due to hypoventilation, where we're not breathing it off fast enough. Metabolic may be due to elevation in any non-carbonic metabolic acid, lactic, phosphoric, keto, and that's a little bit harder to untangle. Is the pH high? Are we going in this direction? That's alkalosis. In that case, what's the arterial carbon dioxide doing? Is it low? If it's low, then it's probably respiratory alkalosis, and that's usually due to hyperventilation. We're getting rid of too much carbon dioxide. If it's metabolic, it may be due to a loss of any metabolic acid, for example, through vomiting, gastric suction, a relative increase in bicarbonate. This picture is a little confusing on the bottom part here. It's talking about renal regulation and respiratory regulation, and it's talking about carbonic acid regulation relative to acidosis and bicarbonate to alkalosis, but don't worry about this part down here. Just look at this scale. That's why I use it. But these are initial physiology. Compensations may change that. So we're just looking at the initial change. A compensation may change those. Dioxide may be compensated. Respiratory acidosis is carbon dioxide retention due to hypoventilation. That may be due to respiratory depression, so there's sedation or brainstem trauma, respiratory paralysis, pulmonary edema, so they can't get rid of enough carbon dioxide, so it's retained, asthma, anything that retains carbon dioxide. Any acute airway restriction is going to increase the pressure, arterial pressure of carbon dioxide, because you can't get rid of it. And if you retain carbon dioxide, it's going to go through the carbonic acid pathway, the carbonic anhydrase, and it's going to decrease the pH, because remember that compensation pathway is the CO2 plus H2O to carbonic acid to bicarbonate to HCO3 minus plus hydrogen. So if your carbon dioxide goes up, you're going to go in this direction, and you're going to build up that hydrogen ion. Metabolic acidosis is an increase in non-carbonic acid, so lactic acid, loss of bicarbonate, anything that's not respiratory, basically. For example, hypoxemia, if you're relying on glycolysis, you might increase your lactic acid. Renal failure, you're not filtering out enough of the, you're filtering out too much bicarbonate and you're not replacing it. Diabetic ketoacidosis. We'll talk about metabolic acidosis a little bit more later when we talk about anion gap. Acidosis compensations, the cellular proteins can buffer and bond hydrogen. We saw that a few slides ago. We've seen before a few times that hydrogen can move into the intercellular fluid in exchange for potassium if there's acidosis, but there's that hyperkalemia risk. Respiratory acidosis specifically, if it's chronic, you're going to have to do these hydrogen compensations, the building the ammonia and the phosphate, getting that hydrogen out to get that urine pH up. Metabolic acidosis compensations, if it's metabolic acidosis, your primary compensation is going to be respiratory. So with metabolic acidosis, you want to get rid of the carbon dioxide. So you can move it on the carbonic acid system towards the carbon dioxide, getting rid of that. And so the hyperventilation will be stimulated to get rid of some of that carbon dioxide to increase the pH. And long term, there can be the same deal here with the urine, the renal secretion of hydrogen. Respiratory alkalosis occurs with an excess loss of carbon dioxide. With the excess loss of carbon dioxide, it reduces carbonic acid and increases the pH. So for example, hyperventilation, if too much carbon dioxide is lost. Altitude hypoxia. What happens with altitude hypoxia is people feel like they're not getting enough air, so they'll breathe more. Well, the thing is, they're not getting any more oxygen because they're at a high altitude, but they're losing carbon dioxide. So they're actually going to generate a respiratory alkalosis. Panic attack, improper medical ventilation can do those. Metabolic alkalosis, a relative increase in bicarbonate concentration because you're losing metabolic acid. So maybe vomiting or gastric suction, you're losing acid, so your bicarbonate becomes relatively more elevated. Compensations for alkalosis, there's that shift of hydrogen again in exchange for potassium. In this direction, though, we're going to take the hydrogen out of the cells to try to increase it in the extracellular fluid, but that means that potassium is going to go into the cells, so there's a hypokalemia risk. Cellular protein buffers will release hydrogen to try to increase the hydrogen concentration. Respiratory alkalosis compensations, you're going to get the renal compensation. Reduced renal secretion, so the opposite of what we saw on the last slide, and reduced bicarbonate reabsorption. So we're not going to push that out into the renal cells. We're not going to want to get rid of that hydrogen or absorb that bicarbonate. We want to compensate by reducing that mechanism. Metabolic alkalosis, if it's metabolic, your compensation is going to be respiratory. So you're going to reduce your ventilation rate and depth because you want to increase your blood arterial carbon dioxide to decrease your pH. You can also have a renal retention of hydrogen there. Objective 7, describe what is meant by the anion gap and explain the significance of the abnormal anion gap in metabolic acidosis. The anion gap is a measure in extracellular fluid that measures unmeasured negative ions or anions. It's a gap because if we included every charged particle, the equation would be 0 because it would be neutral. How many are not accounted for, though? That's what we're trying to look for. It's used to try to determine the cause of metabolic acidosis. Different metabolic acid increases will impact the anion gap differently, and you'll learn more about those clinically, but I want to explain what it actually is here. So when you hear about it, you'll kind of know what you're looking at. What the anion gap looks at is it looks at the cations, and we subtract the anions, and we look at how much of a gap there is. What's the difference between the cations and the anions? And it can be, it can vary. That number is going to vary, but in this example, it's 10 milliequivalents per liter. It's positive because we are accounting for more anions than we are cations than we are anions. Look at this. If we look at this equation again here, we can get different kinds of information from the equation. So let's say hyperchloremic metabolic acidosis. Hyperchloremic metabolic acidosis is going to maintain the ion balance, because what happens is in this condition, if you lose bicarbonate, you may balance it by retaining chloride and you don't change your gap. So you don't change your gap, you've got metabolic acidosis, but you don't change the gap then. But there are metabolic acidosis that do change the gap. They elevate the gap, which means they make the gap bigger. So you're not accounting for, you're accounting for fewer anions because your gap gets bigger. So your number will get higher when your gap gets bigger. So you're accumulating anions other than chlorine, and those anions are not counted in this equation. So for example, methanol ingestion metabolites will generate anions, lactate anions, acetoacetate from alcohol, sulfates, phosphates, any of those anions are not counted in this equation, and if they're there to neutralize the cations, your gap is going to get bigger because you're not counting them. You're not counting those anions. Increased keto acids in diabetes, that will bind to the bicarbonate, and so it will give a bigger gap because that takes bicarbonate out of the equation. Not all of it, but it will bind to it, and so the gap gets bigger because the bicarbonate goes down. And if the chloride doesn't go up to compensate, your gap number is going to get bigger. A low anion gap may result if plasma proteins are lost. Plasma proteins carry a negative charge. So if chloride goes up to compensate for the loss of the proteins aren't measured, so chloride will go up, and that's going to make your gap between your positives and your negatives lower. You're going to have a smaller number, smaller gap number for that. All right, last slide. 340, rocking. You cut almost 25 minutes off of this. Don't worry, you're not losing any information though. It'll come back in later chapters where it fits better. Exam 1 only covers material from these 2 chapters. It's not for a while. Don't panic. It's, I think, the week of Memorial Day, so it's still a little ways away. It's a Wednesday. The exam questions will involve interpretation of the material covered in the lecture notes and in Collaborate sessions. The exam questions rarely involve material from the book that was not covered in the notes, but the questions do involve understanding the material rather than memorizing. So I am not going to take things word for word from the notes. There's not a point to memorizing these notes. That will not help you. You've got to understand it. I can come in sideways, backwards, from the top, from the bottom, and I can make sure that you're understanding by taking the components and coming in the middle and making sure that you understand what would happen as a result of that. So don't just memorize these notes, because that's not going to help you. It's time to move beyond memorizing into fully understanding the information, and that's going to involve integrating it in a very active learning way. And we'll talk more about the systems diagrams in when you're here for orientation. But I gave you this early to try to alleviate some of that anxiety so that you can kind of spend a little time with it this week. And I don't expect you to have it fully known and expert level by the time you get there for orientation, but I do want you to be familiar with it because most of it should be review. Some of it I know is probably not, unless you've had advanced cardiac, some of that ECG stuff, but again, that's not the focus. The focus is the basic physiology of cellular biology. So that's what we're going to build on as we go through. I'll talk more about the discussion areas when we get to campus. So you won't be able to even access Blackboard until you get to orientation, so don't worry about this. What you need to do is go through these, through the lecture, through the notes, and then have those muddiest points, like I said in the email, ready, and then give those to me Monday or email them to me ahead of time, and we're going to talk about them on Tuesday. So I need at least a little bit of time to prepare. To see where the sticking points are. All right, I'm looking forward to seeing you next week, and we'll have some fun with physiology, and it will be, don't worry, we have a lot of fun. I'll see you next week.